Hello. Welcome everyone to the 158th annual meeting of the National Academy of Sciences. I'm Marcia McNutt, President of the Academy, and it's my pleasure to welcome both members and anyone just interested in science to my annual address. This is my opportunity to review the latest projects and activities of the National Academy of Sciences to keep you informed and to urge you to engage in fulfilling our mission. It was just 1 year ago that we had to quickly move an already planned in-person annual meeting to a virtual format. At the time, I could not have imagined that 1 year later, we would still be in the throes of a global pandemic and unable again to meet in person. And therefore, before I begin my report, I think it fitting to spend a few moments reflecting on the more than 570,000 Americans and 3 million people worldwide who have lost their lives to this deadly virus. I note that the US has 19% of the deaths while only having 4% of the global population. The graph you see here was recently published in The Guardian. It compares the US death toll from disease, war, and accidental deaths, both pre-COVID on the left and COVID on the right. It is important to put this death toll into perspective. Although heart disease and cancer remain the #1 and #2 most common causes of death, COVID-19 has now risen above even the horrific yearly death toll from major wars, and well above other frequently cited causes of death, such as guns and traffic accidents. It is now the 3rd leading cause of death in the US. So everyone, please join me now for a moment of silence in remembrance of all of those souls lost. Thank you. And now we'll move on and I'll highlight some of the great achievements here at the Academy in the past year. As I did last year, I've organized my presentation following the template of the Academy's strategic plan. The first goal is to apply science for the benefit of humanity. And of course, there's been no more urgent need to apply science for the benefit of humanity than to address the pandemic. The National Academies quickly assembled a standing committee to help inform the Health and Human Services Assistant Secretary for Preparedness and Response and the White House Office of Science and Technology Policy on critical science and policy issues. The standing committee was chaired by former National Academy of Medicine President Harvey Feinberg, and it includes 27 experts in emerging infectious diseases, public health, crisis standards of care, risk communication, and regulatory and legal issues. The committee established a new product called Rapid Expert Consultations, which are written by the committee and subject to accelerated review by the Quality Assurance arm of the National Academies, its Report Review Committee. They represent the best evidence available to the Standing Committee at the time that each publication was released. These rapid expert consultations demonstrate the capability of the National Academies to act swiftly as the current crisis demands. They also serve as a proof of concept for delivering advice on the timescales that leaders need to take action. These novel rapid consultations have covered urgent questions such as crisis standards of care, diagnostic testing, social distancing, virus seasonality, and allocation of monoclonal antibodies. The academies have released 2 short consensus reports, including one on genomic epidemiology data infrastructure needs for SARS-CoV-2, and that report was chaired by our Academy Vice President, Diane Griffin. As of February 2021, These rapid expert consultations have been downloaded more than 50,000 times and read more than 90,000 times across the US and the world, and they've been subject to more than 60 news articles. Another very high-profile COVID activity was the formulation of a framework for equitable allocation of vaccines. A committee was convened last July in response to a request from the National Institutes of Health and Centers for Disease Control and Prevention. Its final peer-reviewed report was released in less than 3 months in September 2020. The framework's ethical and procedural principles underlie recommended allocation criteria and phases of distribution. The framework used the US Census Social Vulnerability Index to identify communities that are in need of special help. The report recommended the immediate allocation of 10% of the US COVID-19 vaccine supply for distribution to countries in need, with an ongoing commitment informed by global need. In terms of impact, these reports have been downloaded 21,000 times and viewed 32,000 times since the release. The Kaiser Family Foundation found that almost every state plan for priority distribution of vaccines relied heavily on the guidance from this report's recommendations. National media highlighted the relatively broad consensus among public health experts about the report's allocation framework. Now, you recall at last year's annual meeting, our symposium focused on the idea of creating a set of grand challenges, civilization shaping ideas to inspire researchers and the next generation of scientists. One speaker from each academy disciplinary class put forward an idea for a grand challenge. Early this year, we began convening a series of virtual meetings to gauge our members' interests in pursuing these topics, along with a few other ideas that came up from the membership. All of the online discussions to date have been excellent. Members considered what about the challenge makes it grand, and what could the National Academy do to advance each of these potential challenges. 2 of the meetings addressed the UN Sustainable Development Goals, including how best to expand demonstration projects to scales that actually matter. Discussions at other meetings explored the search for life beyond Earth, sequencing Earth's biogenome, and diagnosing, treating, and preventing human diseases. But several of the grand challenge topics cut across all the others, and they are highlighted in red on this slide. In other words, it's hard to imagine that we'd be able to achieve any of the proposed grand challenges unless we simultaneously forge new paths to science literacy, better integrate the social, biological, and physical sciences, and become a more equitable and inclusive community that embraces the diversity of America. All of these topics involve creating the talent pipeline, research culture, and public support that will be needed to pursue any scientific grand challenge. I'd also like to point out that the fascinating discussion of a grand challenge in synthetic biology, which I've highlighted in blue here, was also in some ways cross-cutting. Although harnessing and ethically pursuing synthetic biology is a grand challenge in itself, it also can be considered a tool for addressing health and sustainability goals. We will complete our conversations with NAS members next month on these grand challenges, and then start planning what an NAS grand challenge program might entail. Now, some of the themes in the Sustainability Grand Challenge will be addressed in the first ever Nobel Prize Summit, which the National Academy is hosting in partnership with the Nobel Prize Foundation, the Potsdam Institute for Climate Impact Research, and the Stockholm Resilience Center Beeger Institute. The summit will address 3 themes: climate change and biodiversity loss, reducing inequality, and third, technologies with the power to transform the way we live and work, including of course, the intersections of these three topics. The summit will be presented over 3 days to an international audience with a 2-day main stage program for a broad public audience, supplemented by scientific technical sessions and separately hosted partner sessions on the third and final day. We have more than 23,000 people registered for it, and that was as of last Tuesday. We look forward to welcoming attendees from across the globe when we open our virtual doors to what we hope will be the first of many productive events organized in collaboration with the Nobel Prize Foundation. Now, holding a major international summit is one way that the Academy can advance a grand challenge. Another is by producing a consensus report that defines the research needed to address that challenge. A relevant example is this recent report on space nuclear propulsion for human Mars exploration, which evaluated both nuclear and nuclear thermal propulsion. Both technologies will require a significant investment now to meet a proposed 2039 launch date. Another way that the Academy can advance grand challenges is by creating cross-cutting initiatives. We are seeing the impact now of changing climate across the country, with implications for our coastal communities, our health, and our personal and national security. The Biden administration has declared tackling climate change as one of its top 4 priorities. And just 1 week after taking office, President Biden took initial action on this priority by issuing a sweeping executive order. In response, the national academies are ramping up their efforts across all of our divisions to inform the development of climate change priorities. This cross-directorate program will address the human health, the equity, the ecosystem, the economic, the national security, the educational, and the infrastructural aspects of climate change. Every day, growing numbers of decision makers at all levels weigh choices affected by the changing climate in developing policies to address it through both mitigation and adaptation. Trusted information is needed to make climate-smart decisions, which require bringing together diverse expertise from the physical, environmental, and social sciences, as well as engineering and medicine. The national academies are well suited to mobilize and coordinate this urgently needed expertise to inform public policy. In early January 2021, the NAS endowment provided seed funding to launch a new climate program to meet the scale, scope, and complexity of the problem. This initiative can address the difficult problem of optimizing sometimes conflicting priorities to achieve an equitable, sustainable, and resilient climate future. Our aspiration is to multiply the impact and value of our work across the spectrum of Academy's activities to a range of influential audiences, and to ensure that we are able to be nimble and responsive as new challenges arrive. I ask anyone who's interested in helping with this timely climate initiative to drop me a note and let me know of your interest. Now, just to show you the vast array of authoritative reports we already have in hand that will provide the bedrock of information to support this new initiative, let me quickly run through some of the past year's consensus reports relative to addressing climate change. So this report on reflecting sunlight is one example of the products that will be part of the new initiative's portfolio. This was chaired by NAS member Chris Field. The Climate Change Intervention Report that I chaired earlier in 2015 called for creating a framework for the governance of research to advance understanding of the potential impacts of actions to reflect sunlight back into space in order to mitigate global warming. This new report affirms the earlier report's findings that solar engineering is not a substitute for reducing greenhouse gas emissions. And it calls for a small national research program to address potential risks and weigh the benefits. It proposes a transdisciplinary research agenda while providing a framework for domestic and international governance of this research. Another report that helps to show the range of topics that need to be embraced by the new climate initiative is this one on decarbonizing the U.S. energy system. The committee was asked to evaluate the status of the technologies, policies, and societal factors needed for decarbonization, and to recommend research and policy needs. Its first report focuses on near and midterm high-value policy improvements, research investments, and approaches required to put the U.S. on a path to net zero emissions. A second report is expected in 2022 that will assess a wider spectrum of technological, policy, social, and behavioral dimensions of deep decarbonization and the interactions between those aspects. In order to reach net zero carbon by 2050, the United States must act now to accomplish its technical goals of producing carbon-free electricity, electrifying energy services in transportation, buildings and industry, invest in energy efficiency and productivity, to plan and permit building critical infrastructure, and finally, to expand the innovation toolkit. Done right, decarbonizing our energy system could strengthen the U.S. economy, promote equity and inclusion, support communities, businesses, and workers, and of course, address climate change. Two other important climate-relevant energy sector studies include one on the electric power grid and a second on vehicle fuel economy. In the report on the future of the electric power, the committee assessed how the grid might evolve in response to advances in new energy technologies, changes in demand, and future innovation. The committee provided recommendations to ensure that electricity service remains clean and sustainable, reliable, and resilient, while facilitating innovations and introduction of disruptive technology, policy, and business models relevant to the power system. The committee another report addressed light-duty vehicle fuel economy. They identified opportunities to improve the efficiency of internal combustion engines, hybrid and battery electric engines, and fuel cell vehicles in the 2025 to 2035 time period. It concluded that zero-emission vehicles are the future of vehicle energy use and emissions reductions in that time frame. Statutory authorities and fuel economy regulations need to be updated to accommodate advances in zero emissions vehicles, as well as automated and connected vehicle technology. To prepare for a future of zero emission vehicles, we need to overcome consumer barriers and current infrastructural constraints. So climate is not the only topic for which we are exploring new initiatives. Deferred maintenance, delayed replacement, and indefinite reliance on piecemeal temporary fixes has undermined the resilience of U.S. infrastructure to both environmental and other stressors. And any solution to this problem must account both for changing climate and opportunities for climate mitigation. Inattention to U.S. infrastructural renewal is at best holding back the full potential of our economy, and at worst, resulting in deaths and other harms to the people of the United States. Under President Biden, the nation may be embarking on a decades-long, multi-trillion-dollar investment. The scope and scale are so large that it's hard to know where to invest first. So this new infrastructural investment prioritization initiative is a multi-year effort by the academies, and it's built around scenario development, evidence, and engagement, adopting a sector-by-sector approach. So who would benefit from the academies undertaking such an initiative? Well, basically everyone, but especially marginalized and disadvantaged people, low-income communities who are not part of the knowledge economy and are most vulnerable to and affected by climate change and infrastructural failures. But also benefiting are states and counties that are on the front line of climate adaptation, and also large and small businesses that rely on national infrastructure and or cannot provide their own infrastructure. So why would the academies choose to take this on? Well, to address these problems, the nation needs an organization dedicated to the good of the nation, a neutral convener of experts, the public, and decision makers. This must be done by an evidence-based organization that can apply the best science knowledge to the stressors. It also must be done by an organization that understands the technology deeply and looks ahead to what innovation can bring. And it must be an organization with experience translating technical assessment and social needs to practical action. So, of course, that's us. So what would we do? We'd begin with a series of scenario-based activities linking the science on climate action stressors to infrastructure. Then we would examine the safety, utility, and benefits of establishing conditional priorities and identify opportunities that new technologies bring in developing a menu of options for developing and applying a framework to establish priorities. Right now, the Gulf Research Program is planning a pilot project on infrastructure that will help determine what policy questions we should aim to answer. For example, how do we measure cost benefit for an infrastructure investment activity? How do we maximize public benefit per dollar invested in infrastructure? Are there ways to leverage public investment in infrastructure with private investment? And finally, how can we better assure that private assets don't turn into long-term public liabilities? Now, it's impossible to talk about infrastructure without including the Transportation Research Board at the academies, otherwise known as TRB. TRB's activities broadly cover issues of safety, COVID-19, economic recovery, climate change, and equality, all of which are priorities for both the national academies and the Biden administration. Within the past year, TRB has issued a number of reports, webinars, blogs, podcasts, annual meeting presentations, and published papers in each of these subject areas, including several very timely recent reports on equity-related issues. The Transportation Research Board also this year hosted its 100th anniversary annual meeting in January. It was the first meeting of the TRB in its 100 years to be held entirely virtually. The meeting had a record 20,000 participants from around the world, with over 400 sessions and over 400 committee meetings. Mr. Dorvell Carter, Jr., who's president of the Chicago Transit Authority, gave a compelling plenary lecture about equity impacts in public transportation. And I had the privilege to be the keynote speaker at the chair's plenary session. Now, part of our goal in providing science for the benefit of humanity is to support international programs as well, especially ones that are promoting sustainability. The NAS has been asked to coordinate the U.S. contribution to the UN Decade of Ocean Science for Sustainable Development to create the science we need for the ocean we want. We have created a robust web platform for news and research updates from more than 60 organizations, including businesses, research institutions, professional societies, and NGOs. We've received 90 submissions of ocean shots, which are like moonshots, but for the ocean. They're transformative ideas. We are hosting ongoing dialogues to nurture ideas and programs. 80 scientists have already applied to become early career liaisons. We created a youth advisory council and a Draw Your Decade contest for children. The U.S. meeting that launched this initiative in February of this year drew more than 1,200 registrants and featured 80 ocean shot posters and 13 other exhibits. Now, Academy members often ask me whether our reports have impact. Well, here's a good example. A Roadmap to Reducing Child Poverty was a consensus report that was released in 2019, so 2 years ago. In those 2 years, it's been downloaded 40,000 times by researchers, policymakers, practitioners, and advocates around the globe. It's been cited in 66 journals and publications and has been featured in more than 40 articles and opinion pieces in major media outlets, including NPR, the New York Times, Vox, The Economist, and CBS, among others. The report provided the evidence foundation for the $3,000 child allowance in the American Rescue Plan of 2021. It was presented more than 20 times to congressional staff and to research and policy-focused organizations. It was incorporated into the election platforms of 6 presidential candidates. Okay, so now let's move on to goal number 2, which is to improve public understanding and appreciation of science. While we have initiated many activities under Goal 1 that apply science for the benefit of humanity, it's only by improving the public's understanding and appreciation of science, the topic of this goal, that we will maximize their impact. Last year, I introduced to you our Based on Science project, which works to counter misinformation on the web by providing evidence-based answers to common questions in accessible language. This past year, Based on Science has really taken off as citizens eager for trusted information about COVID use it as a resource. We have exceeded 5 million impressions per month with this content on the web, and the website is actually driving traffic to other Academy products and information. It has the top ranking in Google searches and has now been translated into Spanish as well because the demand was so high. Now, as part of helping to make our work accessible, one example I show here is how our communications department has taken our report on reproducibility and replicability of science and leveraged products to inform the public about how science advances. The rollout included synopses of the report's findings and recommendations that was targeted to professionals, but it also created videos and social media content that was targeted to the public. And there's also a Based on Science posting describing how repeating experiments is part of the science quality control system, and that's targeted at the lay audience. Now, one of our most successful outlets for delivering science to the public is our Science and Entertainment Exchange, which links science experts with Hollywood film and television professionals. Since its launch in late 2008, the exchange has facilitated more than 3,500 technical consultations between Hollywood professionals and scientists. It also produces a series of lively events to inspire creative thinking, such as a salon that featured NAS member George Church, who spoke to a high-level audience of entertainment industry executives about the emerging science of CRISPR. In March 2021, Deadline Hollywood reported that producers plan to work directly with Dr. Church on developing a dramatic television series featuring synthetic biology. In October 2020, LabX, which is the public outreach program that's the successor to the Koshland Science Museum, initiated a competitive COVID-themed game contest to encourage pro-social pandemic behaviors. Bringing together gamers, designers, and public health experts from around the globe, the event, which was called Jamming the Curve, attracted more than 400 participants across 6 countries who collectively created more than 50 COVID-themed games in the short span of 2 weeks. The Jamming Your Curve event received national media attention, including a feature story in the LA Times. Now, the winning game out of those 50 is called Cat Colony Crisis. and it challenges players to contain a mysterious outbreak on a spaceship full of rambunctious kitties. The judges chose this game because it conveys the importance of individual behaviors in stemming the tide of a pandemic in a lighthearted, easily accessible manner that avoids being overly prescriptive. The game became publicly available just on February 9th, and it's free to play online. It's now been downloaded in that short time more than 50,000 times, and it's been so popular in Mexico that we had to translate it into Spanish. The game has just been nominated for a Game Development World Championship Award. Let me turn now to the work of the Academy's first ever artist in residence, Chris Michael. He's creating a photo documentary of how science, engineering, and health professionals have responded to the impact of COVID and how it has affected their work. The photos will appear in an online exhibit. Here's an example of a photo shoot with Bob Wachter. Now let's go on to Goal 3. Goal 3 is to improve the culture and practice of science, including the reward system and the culture of research. We know that mentorship is key to the development of everyone in STEM, but few professionals have had the opportunity to learn how to be a really great mentor. In this 10-part series of podcasts, we present the personal mentorship stories of leaders in academia, business, and the media in their own words. The series teaches evidence-based mentorship practices that can develop the skills needed for the most effective STEM mentoring relationships. The series highlights the effective practices of Academy members, such as NAS member Jim Gates and Keith Yamamoto. Following the 2018 release of the landmark Academy report on sexual harassment in academia, our staff formed an action collaborative, which is a coalition of the willing, to implement the report's recommendations in educational institutions of all sizes and types. The collaborative includes more than 60 higher education and research organizations working to identify, develop, implement, and share approaches and practices that can prevent and address sexual harassment. Last year, they shared more than 100 policies, practices, or approaches to implementing the report's recommendations, and they'll continue to enable higher education and other organizations to learn and build on this work. Many prominent institutions have already used the report and the help of the Action Collaborative to examine their institutional systems and policies and develop implementation plans. They have cited the report when announcing or explaining changes to their institutional policies and practices. The report led to the work of the Society's Consortium on Sexual Harassment in STEM. This is a group of over 100 scientific, technical, and medical societies working to address sexual harassment in all its forms and intersectionalities. Building on the report and the committee's recommendations, NSF, NIH, and NASA have all made policy changes, revised practices, or made statements referencing the report. The report was referenced in 4 pieces of federal legislation and other letters or reports from members of Congress or congressional committees. The report also informed legislation in Washington state that was passed in spring 2020 to address the issue of pass the harasser among higher education institutions. The report also inspired the documentary film Picture a Scientist that was just broadcast on PBS's Nova across the US. It has been mentioned in well over 100 domestic and international media outlets and continues to be cited in major media articles now 3 years after its release. The report has been downloaded 31,000 times and read online more than 150,000 times. It is ranked number 5 in impact by Altimetrics among all Academy's publications ever. This past year, we also released a new report to address the underrepresentation of women, particularly women of color, in STEM disciplines. The committee sought out institutions that were doing much better than others in hiring and retaining women, discussing examples of successful strategies that others can follow. Last year at this time, I reported on the results of a Sunnyland retreat to explore implementation of the recommendations from now several academies' consensus reports that what is needed is a body that would align incentives in the research enterprise with the norms of science. What's also needed is a body to coordinate sometimes conflicting policies across the entire research system, from research funders to execution to publication and eventual incorporation of those results into public policy. Based on the report from that retreat, the Academies is stepping up to launch an activity called SCORE IT. And SCORE IT is an acronym for the Strategic Council on Research Excellence, Integrity, and Trust. The activities of SCORE IT will include promoting the study of effective practices, developing new resources, distinguishing systemic problems from outliers. Here I've listed on this slide some topics that are ripe for SCORE IT to tackle immediately: conflict of interest, the academic reward system, open science practices, and student training and mentoring. And now for a change of pace. The cultural programs activity of the National Academy of Sciences, with input from our Committee on Women in Science, Engineering, and Medicine, has developed a new exhibit to raise awareness of the contributions of women in STEM and to increase the representation of women in images displayed in the NAS building. This inaugural collection features 40 women reflecting diversity in age, race, and discipline. They are all pioneers in science, engineering, and medicine who have broken barriers and made significant contributions to their fields and to American society. Others will be added to this inaugural group as the exhibition is curated over time. The exhibition was launched online in March during Women's History Month and has attracted more than 2,500 unique visitors and 11,000 total page views. The physical exhibit will be installed in the East Gallery when the NAS building reopens, likely in the fall. So with that, I want to say a final word of thanks to all of you out there who volunteered for committees, for reviews, for awards, reviewing PNAS, served on our boards, and other activities. And until next year, I wish that you all will stay well, and I hope that we'll be able to meet in person in the coming months. So thank you very much for your attention. We now have a break scheduled until 12:30, when we will all reconvene for this year's symposium on COVID. So thank you all very much. So long.
Welcome everyone to the annual meeting scientific symposium, Strategic Planning for Crises, where we stand with COVID-19. I'm Diane Griffin, and I'm the Vice President of the National Academy of Sciences and chair of the Council Committee on Scientific Programs, which put together this symposium. So the overview of what we're going to talk about this afternoon, or whatever time period you're in for the next few hours, actually, is that, first of all, I'll give a quick overview of some of the issues and some of the basic science underpinnings for the current status of the field. Then there will be a panel on the virus, its evolution, replication, and disease, the response to the pandemic, the mitigation strategies, predictions, the vaccines, the development, efficacy, and delivery of those vaccines. And then the last session will be on resilience and recovery and how we can do better next time. This is a curve that I think all of you are familiar with, which are the number of cases in the United States. And then we have a substantial share of the global burden of cases, which have now come down, but are still, transmission is still occurring at a substantial level. This is being counteracted, though, by the deployment of vaccines, and this shows the numbers of people in the US now that have been vaccinated, and more than half of the adults have been, and we're on a steady upward trajectory. So this remarkable success really of initiating vaccination within a year of actually identifying the virus has come on the shoulders basically of a number of people who have worked in this field for a long time. And I just want to highlight the fact that it wasn't all started a year ago. It started decades ago in an understanding. First, I'm going to highlight two different aspects of this. First is the understanding of the coronavirus and the function of the spike protein. And this was research that's been done on viruses that were not human viruses. They affected mice, pigs, chickens. And in the 80s and 90s, a lot of the basic understanding of the coronavirus spike was accomplished. And then the second is the mRNA. It's a new vaccine platform, but people have been working on the basics of what you needed to have for an mRNA vaccine, again, for a couple of decades. So just to point out these two areas, and there are many other contributions that we could spend time on as well. So first of all, the spike, it's a little different picture of the coronavirus than you often see with the computer graphics, but this was the electron microscopy, and this is the way that viruses were originally grouped, was by having these very prominent spikes on the surface. And one of the things that was recognized with the early studies of coronaviruses was that often they were quite host specific, although we'll be talking about how the virus can move between species, but it was recognized that the spike was a determinant of their this kind of specificity. So in the first thing I'd like to highlight are the functions of the spike protein. First of all, recognizing Kay Holmes, who did a lot of this early work on showing that the glycoprotein, that spike protein of coronaviruses, first of all, had to be was responsible both for the attachment and the entry of the virus into cells, and that proteolytic cleavage of that spike was required for it to have the self-fusing activity. So it required a cell function to actually perform that cleavage if the virus was going to be infectious. And another aspect of the research on the spike was the first identification of a coronavirus receptor, which was for mouse hepatitis virus. So this work was done with a model system. And and identified this carcinoembryonic antigen as a receptor for mouse hepatitis virus, and then showed that it was species specific. So the same protein from that served that same function for other species actually did not bind to the spike. So now we know more about that, and this is a monomer of the spike. And here's where the MHV receptor binding occurs, and there are two areas of the spike, as you can see, that project and can serve as receptors. So it turned out that the one that the mouse hepatitis virus uses, the N-terminal domain, is different than what we now know for SARS coronavirus, which is the C-terminal domain of this spike protein. But we also know that for SARS, that many of the mutations that are occurring in the N-terminal domain, and that monoclonal antibodies that are protective bind to both of these two domains. So understanding that has become important as well. So this is an overall picture of the spike and the conclusions from that. There's these two regions on this linear version of the spike protein that can bind either to the mouse hepatitis virus receptor binding domain, or the N-terminal domain, or this other region that can bind to the SARS ACE2 receptor. But both regions are important for the function. And the fact that we now know there's two cleavage sites that are required for the spike protein to be infectious. The second area I want to highlight was very important for development of the mRNA vaccines. And that was really understanding better how the cell knew whether an RNA was a foreign RNA, like from a virus, or was its own messenger RNA that needed to be translated to make cellular proteins. And so these two investigators were studying this really basic aspect. So Catalin Carrico and Drew Weissman were studying this basic aspect of RNA recognition and how the cell differentiated foreign from self RNA. And what they showed was that that host RNA had structural modifications that foreign RNA didn't. Methylation and the pseudouridine were two of the most important. That the absence of these modifications was what induced these innate inflammatory cytokines. So, and that then modifying the RNA so it looked more like host RNA would make it less reactogenic. And this is just one piece of data for tumor necrosis factor, which is an inflammatory cytokine, showing that unmodified RNA induces a lot. While if you add these modifications either in combination or singly, that you greatly reduce the amount of these inflammatory cytokines that are produced. And then the next discovery or next important thing to know about these RNAs was that, that putting these modifications into the RNA actually improved the translational capacity and stability of the RNAs and therefore could be incorporated into the RNAs that are going to be synthesized to make vaccines, although initially the reason for doing this research was not that. So this is just diagrams of this pseudouridine version of uridine and the regular version of uridine, which is then recognized as being potentially foreign, and the fact that the base pairing is not different for these two modifications. However, translation of an RNA that has this pseudouridine modification is actually much more efficient. So here it is with the modification compared to the unmodified RNA. So by adding in these modifications that were identified, you could not only make the RNA less reactogenic, you could also make it translated better. So important improvements that made the RNA potential a reality. So the overview of the symposium for today is to look at who you're going to hear from. Is first of all, the first panel is chaired by Linda Safe, and we'll have Ralph Barrick and Lisa Ng. The second panel, it'll be chaired by Bill Press, the treasurer of the NAS, and will be include Nicole Lurie, Jenny Durkin, Lauren Myers, and Caitlin Rivers, and each of these will be introduced at that time. And then the vaccine session with Reno Rappuoli will feature Barney Graham, Larry Corey, and Amanda Cohn. And then lastly, the solutions to all these problems, Marcia McNutt will chair that group with her panel of people that are working on scenario planning for these kinds of emergencies. Karen Dinan, Gary Michaelis, John Lumpkin, and Janet Napolitano, and Martin Jose Sepulveda. So the overall plan for these sessions is that there will be a 10 to 20 minute panelist videos are available now, and they'll be available later. So essentially all of the panelists, most of them at least, have recorded these videos, which will be much longer versions of their background information. But each one of them is going to be able to give a short version as a part of the panel discussion, so you have an idea of what those videos contain and can identify the ones that you want to go back and watch if you haven't done it. Each panel is going to meet for 50 minutes to basically do the short presentations and then have plenty of time for discussion and questions. We have a 10-minute break between the panels, and you can submit questions for the panelists that will be incorporated into the discussion. So that's the overview, and the first panel is going to be chaired, as I mentioned, by Dr. Linda Safe. So Dr. Safe is a distinguished university professor in veterinary preventive medicine at Ohio State University. And she's organized this panel. She's been one of the pioneers for work on coronaviruses, primarily, again, from the point of view of these viruses as veterinary pathogens, which they are definitely been major areas, and has worked on developing vaccines that have also been tested, and also were understanding those vaccines has been important for the development of the SARS coronavirus vaccine as well. So I'm going to turn it over now to Dr. Safe for her panel. Thank you.
Okay, so I'd first like to thank Diane for the nice introduction to set the tone for this symposium, but also the National Academy of Science for organizing the symposia. So I'm going to, what we're going to do in this panel, this is an overview of the coronavirus, emphasizing COVID-19, the SARS coronavirus 2. And we're first going to have an overview with 2 slides from each panelist that will have a summary of their pre-recorded talk, and then another slide that's going to have some of the gaps in our knowledge related to that focus area. So I'm first going to present my summary slide and overview related to my talk. And so the summary is shown here. What we can see is my talk focused on interspecies transmission of coronaviruses, and in both humans and animals, and something about their origin. So this summarized the ecology and chronology of coronavirus emergence, with the chronology down here. So the bats and birds are believed to be the natural host reservoir for all coronavirus, both the mammalian and avian coronavirus. We have the 4 genera, alpha, beta, gamma, and delta coronavirus, with these latter 2 from spillover from avian host reservoirs, and the alpha and beta, the mammalian coronavirus, from bat host reservoirs. So I'm going to focus on the zoonotic transmissions, but you can see many, many species are susceptible to these coronavirus, both mammalian and avian species. So in terms of the coronavirus transmission in the alpha coronavirus, we had the spillover of 229E, and this was thought to have occurred in the chronology down here by molecular clock analysis from bats through alpacas to humans in the 17 to 1800s. But predating that, we had one of the earliest coronavirus in spillovers from bats, potentially directly to humans in the 12 to 1500s. And then in the beta coronavirus, the Imbeco subgenera, we have the spillover from cattle of the OC43 to humans. This was thought to have occurred in around 1898. And it's very interesting because at that time, there was a worldwide pandemic. It was called the Russian flu or Asian flu, but the cause was not actually identified. So some people believe that scientists believe that this actually could have been the first coronavirus pandemic that occurred with this spillover event. So this shows we have the 4 existing alpha coronavirus, 2 with the common cold, 229E, NL63, and then 2 beta coronavirus associated with common colds that are endemic, the OC43, HKU1. So going on to other zoonotic spillovers more recently for the beta coronavirus, the Sarbeco coronavirus, the original SARS coronavirus transmission, and then about 20 years later, almost, the SARS coronavirus 2 transmission, and then the Merbeco virus, the spillover from bats to camels of MERS. Now, both of these, the SARS coronavirus 2 and the MERS, are ongoing. So we have the pandemic, pneumonia, some fatal strains, all beta coronavirus, SARS, SARS coronavirus 2, and MERS. The chronology is that the SARS coronavirus occurred in China in 2003, spread worldwide, spillover from bats through civet cats and other small carnivores in the wild animal markets. And then about a decade later, we had the origin of the Middle East respiratory syndrome coronavirus in the Middle East from bats through camels to humans, and this is ongoing in the Middle East. And then most recently, the SARS coronavirus 2, again, likely a bat host reservoir, spillover either directly to humans or through an intermediate host. But the other point I want to make, there are ongoing and prior spillovers often from wild animals sourced from birds or bats of coronavirus into livestock, and particularly focusing on swine. So the porcine delta coronavirus in 2012, a bird source, an avian reservoir, and then these swine acute diarrhea syndrome in China, southern China, from bats in 2017. So what the problem is, or a major concern now, we know that we have reverse zoonosis with the SARS coronavirus 2, with spillover now from humans into multiple species. The most worrisome one is probably the mink, because there's evidence that once the virus is transmitted from humans to mink, it spills back over from the mink to the humans. We also have transmission, natural transmission from humans to pets in the household, dogs and cats, and then experimental infection of at least 8 species, including various wildlife species, including camelids, including the cervids, the deer, and even the fruit bats. So this is the big concern. If this virus does establish itself in animal reservoir communities, then there's the chance that this will spill back over from the animal reservoir community, where it will persist, recombine, mutate, and potentially spill back over into humans. So going on to the unresolved questions, a number of them, like SARS, did COVID-19 spill over from a bat host reservoir? How was it transmitted to humans? Under what circumstances does interspecies transmission occur? Is there an intermediate host that transmitted the virus to humans? Do aerosols or non-respiratory routes play a role in the interspecies transmission, or foodborne or incidental hosts, pets, wildlife? Will human variant strains be more transmissible or pathogenic in the animals? This hasn't been investigated. All the experimental studies I mentioned were done with the original strain of SARS coronavirus 2. If this continues, will we actually need vaccines for the secondary hosts, such as mink or pets, to prevent evolution of new variants, spillovers, and possible zoonosis back into humans or other species? And then will spillover from humans into animals create these new host reservoirs for COVID-19? And if so, will the animal strains mutate, recombine, and reinitiate zoonotic transmission to humans? And finally, WHO has designated disease X as the next potential pandemic. Will it be another coronavirus? We have several red flags here for possibilities. The SARS-related coronavirus are still circulating in bats, and some of these have been shown to be able to bind directly to the human ACE2 receptor. We have the porcine swine acute diarrhea syndrome, spillover from bats to pigs, and Dr. Berry's lab has shown this virus can also infect human cells. Then we have the porcine delta coronavirus, spillover from avian hosts to pigs, and now there are 3 examples of spillover into Haitian children of these porcine delta coronavirus. So with that, I'll conclude and thank the Academy again. And from there, we're going to go on and to the next person, and this will be Dr. Ralph Berry. And I'll just give a brief introduction because his bio is available online. So he's the University of North Carolina, Keenan Distinguished Professor of Epidemiology in the Gillings School of Global Public Health and Microbiology and Immunology. His research focuses on the pathogenesis and emergence and mechanisms of RNA viruses in terms of host susceptibility, virus genetics, microbiology. And also at the recent Congressional Biomedical Research Congress in early 2020, Dr. Berry updated the U.S. House of Representative members and staff regarding the dangers of the SARS coronavirus 2 pandemic, including research priority and strategies to mitigate risks. So Dr. Berry is a world-renowned career coronavirologist with great expertise on coronavirus and now including COVID-19. So with that, Dr. Berry. You may be muted.
Thank you. I couldn't unmute myself. Thank you, Linda, for that introduction. It's a pleasure to be here to talk. I was asked to review the genome organizations of coronaviruses and to talk about the fundamental transcription strategy of the virus. In that talk, I will mention how flexible the transcription strategy of the virus. I will talk about the presence of cryptic transcription start sites, which allow for the expression and evolution of novel orths or even protogenes in coronavirus genomes. That transcription strategy is actually so plastic that it could also allow for gene duplication events and can be engineered or rewired to make recombination resistant viruses. I'll talk about how that transcription strategy relates to virus evolution and discuss the mechanism of high frequency RNA recombination that can occur during mixed infection of a cell with 2 different coronaviruses. Recombination plays a major role in shaping the genome organization of coronaviruses. And deletions in the spike and the luxury ORFs are common. And this allows for rapid evolution and also change in tropism of coronaviruses from the respiratory to the enteric tract or to other organ-specific disease modalities. In fact, the world record for a deletion in a coronavirus spike is about 150 amino acids in length, described by Mike Buchmeyer a couple of decades in the past. Coronaviruses are also unique among all RNA viruses in that they have a specific set of replicase genes that regulate fidelity. This removes mismatches from the 3' end of growing nascent strands. The NSP14 protein that does this job is important for pathogenesis. It regulates resistance of coronaviruses to nucleoside inhibitors. And some data, although speculative, suggest that this fidelity could be regulated such that the mutation rates can change based on environmental settings. And this certainly needs to be studied in more detail. I'll talk about how these three factors regulate the emergence of variants of concern that are currently plaguing human populations across the globe. And then I'll segue into comparative studies that use genetic resources to map susceptibility loci that regulate the SARS coronavirus pathogenesis. This is across multiple members of the SARS coronaviruses, including SARS, bat SARS, and SARS-2. And in those few slides, I will show that there are common loci that are synthetic in the mouse and the human that encode the same cluster of 5 genes that are important for regulating severe disease, and go on to show that one of those genes, CCR9, plays a critical role in protection from severe SARS coronavirus pathogenesis in the mice. So one of the interesting hypotheses from just understanding the basic replication and evolution of the virus is that these variants of concern may be adapting to multi-trait susceptibility loci to subtly regulate virulence during infection, and or use mutation-driven evolution to alter expression of low-level cryptic ORFs, which could enhance pathogenic outcomes. The knowledge gaps in coronaviruses are exciting. We still don't understand the basic mechanisms of transcription regulation of this novel group of viruses. They have the largest genome in nature, and they have the most complex transcription strategy of any RNA virus. And this is ripe for exploration, not only in terms of fundamental knowledge, but also in terms of how to mitigate new strategies to attenuate virus replication and pathogenesis. We know that coronaviruses recombine extremely frequently within a genogroup, but we also know that they can recombine across genogroups. And the frequency of this event, the constraints that regulate cross-genogroup recombination events, is not really known, and this needs to be studied. The entire replicase complex that regulates fidelity is a fascinating topic because it also regulates and has an impact on transcription and recombination frequency. And so understanding that process and understanding how coronaviruses may actually manipulate fidelity function to alter evolution rates would be a fascinating question to study in more detail. It's fairly clear that SARS coronavirus 2 variants of concern will continue to emerge. And one of the interesting features about coronavirus pathogenesis is high virus replication occurs very early in infection, and it's somewhat uncoupled from the high pathogenic outcomes that occur later in infection. This allows the evolution of virulence to be uncoupled from transmissibility. So what will the mechanisms that these variants use to increase not only immune escape, but potentially increase virulence in human populations? What are the host susceptibility loci and the multigenic traits that the virus is evolving to regulate virulence and pathogenesis and transmission? Our own data in the mouse and the human argue that there are common susceptibility loci that regulate pathogenesis across coronaviruses that fall in the three major clades that have been described in humans and animals. How do these genes regulate pathogenesis across species is unknown, and how this contributes to emergence and pathogenesis is still unknown. And then we just still have a fundamental lack of understanding of the mechanisms of pathogenesis of these important viruses. We have a large amount of descriptive human data, but mechanistic understanding, I think, still is beyond our grasp at the current moment, especially in terms of the acute phase disease process, but even more importantly in chronic disease sequelae that occur in multiple organ systems in the human host. And so understanding that process, developing animal models to understand chronic disease sequelae represents a tremendous knowledge gap about these important human pathogens. So with that, I thank you for your attention.
Okay, thank you very much, Ralph. And with that, if you don't mind, stop sharing the screen. We'll go to Lisa Ng. She is the Executive Director of the A-STAR Infectious Disease Lab in Singapore. So we will get some Asian perspective here on the SARS coronavirus 2. She's also Executive Director of the Biomedical Research Council, where she oversees research talent and strategic initiatives. She got her PhD from the University of Manchester Institute of Science and Technology in the UK, studying the molecular virology of coronaviruses. And she's also worked on SARS and the H5N1 bird flu. Her group at the Singapore Immunology Network focus on the immune response to arthropogenic arbovirus, including chikungunya, dengue, and Zika. And then more recently, SARS coronavirus 2 infections and pathogenesis. She's won a number of very prestigious awards, including the Association of South Asian Nations International Young Scientists and Technologists Award in 2008, the Most Inspiring Mentor Award in 2013 at her institute, and she serves in various expert research grants and fellowship committees in the Wellcome Trust and European Union. So her talk was on the infection and immunity of SARS coronavirus 2 from diagnostics to therapies for COVID-19. So she'll provide the summary and the gaps slides. So thank you, Lisa.
Thank you so much, Linda, and also to the organizers for having me here today. So it is indeed an honor, and I'm very happy, even though it is 1 a.m. now in Singapore. I think Linda and Ralph have given the background and also the various key aspects of coronavirus and also the SARS-CoV-2. So my talk really focused on how the various teams both in Singapore and how we also reach out to the other researchers around the world, specifically with the clinical community, to build up this study with the COVID-19 patients in Singapore. And we were one of the earlier countries that started the battle of SARS-CoV-2 as early as late January 2020. So at that time, things were not so clear, except that it was clearly very contagious, and the earlier cases were all imported. So the clinics set up this study called the PROTECT study, which was the aim was to look at the whole chain of clinical questions and also scientific questions from the clinic to the industry. And it was actually written up for Disease X, and that particular protocol was actually written about 4 months before we actually dealt with COVID-19. So it was, I think, the timing. So in my talk, we sort of showed the different categories of the disease severity of the patients, And I think that has been quite well characterized in many countries around the world, which most of the cases were reported to be fairly mild, but there's also a big number that has moderate and disease. And as you can see, with more studies coming up, it's actually more complicated than that. And that also then lead to the very severe cases, the various comorbidities. So the various studies started with very basic questions. Are we able to really identify the specific antibody profiles from the acute patients, and how their profiles actually then change during the course of their disease progression from 3 weeks to a month, 2 months, and 3 months. And you could see from here that in the early days, there have been conflicting reports that the levels of IgM and even IgG were not long-lasting. But we have been having data now to show that actually some of the patients actually do continue to have detectable levels of IgG, even though you could see that there have been some levels of decline. And so this is where we were really very focused on this whole chain, from the clinics, and making use of the various approaches from flow cytometry, proteomics, mass cytometry, immunogenomics, to really understand this entire aspects from antibodies. And we actually showed that we were able to identify specific linear B cell epitopes, and some of them actually could be further developed as serodiagnostics. And as we head on to more than a year, you could see that some of the profiles have also shifted. And by making use of some other aspects of flow cytometry, we also identified the specific immune cells that were enable us to triage patients who had more severe disease compared to the other patients. And we identified key cell types, not just the pro-inflammatory monocytes, but also how neutrophils could actually be used as a severe disease marker. And by looking at the and how some of this could, like I said, be used to strengthen the prognosis. And so we also had a deep analysis of the immune mediators, and how this we showed that clearly, like the 2003 SARS, it is a very pro-inflammatory disease, and you could see from here that the various immune mediators then allowed one to actually move on to study the specific inflammatory pathways, and how this would be useful markers for interventions. And it's also quite clear that in the earlier days, and even up to this point, there have been various attempts to make use of some of the available FDA-approved drugs. And that is an area where it worked for some patients, it did not. So at this point, there's still a lot of questions that is not answered. Perhaps it could be the timing of the treatment that is the most crucial. And also there's a big area of development of monoclonal antibodies, and how some of this potentially still holds some value. And so some of the key questions moving forward, as we are now in year 2 of this whole pandemic, I think it is still quite a lot to be done in terms of really identifying the specific markers of not just protection, but also resistance. And we know that there will be some interesting data coming out from several groups. And Ralph has also alluded to the influence of host genetics. This is an area that that's going to be very exciting. And in terms of severe presentations, I think there are more and more studies coming out on some other aspects of neurological complications, and so some of the specific cell types that are known to be involved. It's not just the immune cells, but actually a whole suite of the various cell types. And once we have a better understanding on some of these mechanisms, then it will open the door to develop possible new immune-based therapies. And again, I just want to close this part where, again, the point that Ralph mentioned, I think the development of more relevant or even better animal models actually would be very enabling to actually understand all these questions that are still very, very unclear. So with that, I will thank you. Back to you, Linda.
Okay, thank you very much, Lisa. So from here, there is one question for Ralph Barrick, and then we can have other questions among the panelists. So the question for Ralph is, what are the major driving forces for the SARS coronavirus 2 recombination, and why have we not seen obvious recombinants so far? Perhaps SARS coronavirus 2 is a recombinant between a bat RAT strain and the pangolin coronavirus. So Ralph, do you have an answer?
So there's been a variety of reports that have argued that SARS coronavirus 2 is a recombinant virus based on a limited number of related SARS-2 bat and animal strains, including the RATG13 and the pangolin virus. There's also a limited amount of evidence that has argued that recombinants are beginning to occur between variants of concern across the globe. So those kind of events, I think, are certainly beginning to happen. Of course, one of the issues with recombination in general with SARS-2 is that what emerged in and around Wuhan, China, was mostly a single lineage that then spread globally. And as the variants begin to accumulate more mutations, now you have more loci available to begin to identify the emergence of recombinant genomes as they occur in humans. We have not seen recombinant viruses between SARS and animal coronaviruses, but I would argue that the bat virome across the globe and the animal virome across the globe in terms of animal species that are now being colonized by the SARS-2 variants with mutations in the receptor binding domain have not been studied at all. And so that's the reverse zoonosis comments discussion point, Linda, that you've mentioned, I think is absolutely critical, because this virus has moved into environments and mammalian populations that it's never seen before, providing all kinds of opportunity for the emergence of new recombinants. And we need to be aware and pay attention to it.
Yeah. So thanks, Ralph, for your answer. I'd like to just follow on with that. You did mention in your talk that you thought cross-genogroup RNA recombination is possible. Do you know of any examples, either experimentally or naturally occurring? And the other follow-up would be, do you think SARS coronavirus 2 could recombine with any of the common cold coronavirus, particularly the beta coronavirus OC43 HKU1, because likely there will be co-infections?
Sure. Probably the most elegant work done on this topic was done in the late 90s, where the mouse hepatitis virus gene spike, the ectodomain, was replaced with the feline coronavirus. The group 2 spike of mouse hepatitis virus was replaced with the ectodomain of feline coronavirus by Paul Masters' group. And that virus was viable, could replicate in culture. And he used this as a strategy to introduce mutations into the mouse hepatitis virus genome by targeted recombination-based approaches. So clearly, recombinants across not only within, let's say, the group 2B beta coronaviruses, but recombinants between group 1 and group 2 coronaviruses are quite possible. we don't know what the constraints are.
Yeah. Thank you. And then go on with a question for Lisa. I don't think we have any other external questions right now, but I encourage everyone to submit their questions on the website. So Lisa, have you been able to identify any biomarkers that are unique to the various comorbidities related to the SARS coronavirus severity? So this would be like the elderly versus people with diabetes versus people with obesity. Is there anything unique to the different populations that in whom SARS is so severe?
Well, clearly, I think there are many data out there that has shown, for patients who have other comorbidities, especially cardiovascular disease, I think these are the group of patients that normally do not fare very well, and also the diabetes. Now, I think in our own cohort, it's a little limited in terms of the disease severity. I mean, this was, in fact, the big bulk of the people were fairly on the mild to moderate, and this was mainly last year. And I think the reason why we had such a big number of cases of more than 60,000 was actually due to the foreign workers dormitory, and these are normally young, healthy individuals. But we have some earlier indications that various the endothelial cells actually play a big role in potentially driving some of these outcomes. And also there are some other studies that are based on the healthy, the younger cohort in terms of severe disease markers. One of the neutrophil cells, especially in the early acute infection phase, actually are very, very prominent. So same as some of the pro-inflammatory monocytes. So I think this is where some of this we could clearly sort of triage the patients by looking at some of these markers. So then in terms of antibodies, it's also quite, there are many cohorts that showed complementary data where interestingly patients who have more severe outcomes actually have higher levels of IgGs and also neutralizing antibodies. So if you were to base on depending on when these patients were recruited, or when you are actually analyzing the levels of the IgGs, I think you could sort of can have some clear idea if these patients might potentially go into the more severe state. So I think that's some of the earlier available information that we have.
Okay, thanks. And following up, I see there's a question for me in regard to the fact that the spike-based vaccines for SARS coronavirus 2 appear to be highly effective, but for some of the animal coronavirus in swine and poultry, they've not been that successful. And what might be the explanation? So we worked long-term for a coronavirus vaccine for swine for the enteric disease transmissible gastroenteritis virus. And we tried a number of just subunit spike vaccines, and some have tried some adenovirus spike vaccines. These were not successful for priming in a naive population. But if we used a live attenuated vaccine for oral priming, and then we could very successfully boost with either a vectored adeno vaccine or a subunit vaccine with just even only the spike. So the situation, the scenario is it's much more difficult to prime a naive population. I think we've been extremely fortunate that the messenger RNA vaccines and the adeno vaccines with just the spike have worked so well in humans. But even in humans, you see that if you've had a previous infection and you give those people the vaccines, they're going to have way increased immune responses, both T cell and B cell responses. So why that difference is, we're not exactly sure. Ralph, you had a follow-up?
Yeah, I have a question for Lisa, if I can ask it now.
Yeah, please.
There have been recent reports that of mortality in post-COVID-19 survivors, and which was significant above control populations. And I guess I have two questions for you. One is, have you seen similar things in Singapore, and would you be willing to speculate on long-term sort of hit and run disease sequelae that might occur, and what the post-COVID-19 pandemic may look like?
Yes, I think that is actually a very good question, Ralph. And well, I think in Singapore we have not quite seen that phenomena that you're describing. But clearly, the long COVID, it is something that is still not very clear. And we are coming to our 12 months, I mean the peak 12 months collection, and just preliminary data sets that we found was in terms of the cytokine profiles. We could not really quite tell any. I mean, they sort of resemble. They have recovered. Most of them have recovered, and just looking at the immune mediators profile, it's not actually very clear. You can't actually differentiate them so clearly. But what is interesting is the antibody profiles, as I mentioned a little bit earlier, and in my talk also you'll get to see a little bit of this, where the differences in the levels, the titers, actually remains different between the patients with a different severity. Of course, a big bulk, I mean, as I mentioned, they have already recovered, and although the levels have declined a little, but actually you can still pick some differences between the patients with the slight more complicated disease manifestation. So that's all I would do, because it is this piece of work. I'm just one of the co-authors in it, and I think the key group, they are trying to finish this part of the work. But I think this is what we have seen. So I don't know if that answered your question, or are you is that something else you are thinking of?
No, I thought that was fine.
Yeah. So maybe this is also, there's several questions from externally, but just to follow up on that, Lisa, do you have any thoughts about the long haulers? The fact that the majority of these are women, very healthy young women. Any explanation, or do you have any biomarkers related to that at this time?
No, I mean, that's something that I actually not able to answer that at this stage. But well, I think this is where if you were to look at some of it could be the some of the immune cells population that might be a little different. But I think again, from what I'm aware of, from what we have been looking at, we are not able to, we don't have that numbers to actually show this. But it will be the some of these elements that might actually have might impact what we are looking, what you are talking about.
Thanks. So another question from externally. What are the possibilities for immune responses selecting for and enriching for the viral variants? Ralph, do you want to start with this one?
I think the epidemiologic data was fairly robust to suggest that, like, the South African variant B1351 and the B1 variant from Brazil occurred in regions where herd immunity was very high. And that would certainly suggest that variants are responding to neutralizing antibodies and escaping some of that protective immunity that's present in people, especially those individuals who have very low neutralizing titers, people who were had asymptomatic infections, or very mild infections that generally have much lower neutralizing titers than those that have severe infections. So variants of concern as they emerge can take advantage of those individuals who had asymptomatic infections and reinfect them. And I think mapping the monoclonal antibody neutralizing sites to the effects on polyclonal responses, some of the nice work done by George Giorgio's group, looking at the serum repertoire response and antibodies that target the NTD loops that have deletions in them, all really make a compelling argument that in certain locations around the globe, variants are emerging in response to herd immunity, and taking advantage then of those individuals who have a low level immunity to allow for reinfection and disease.
And then just to follow up on that, and then Lisa, if you have any response as well. Ralph, you indicated in your talk that you thought these VOCs would continue to emerge with increased virulence, whereas as a virus adapts to a population, usually you expect eventually to have reduced virulence. So why do you think these will continue to have increased virulence?
I think there's an interesting evolutionary theory on the evolution of virulence and transmissibility that increased virulence attenuates transmissibility. Of course, there's obvious virus family members that completely break that rule by causing severe disease much after acute infection, or there's a prolonged disease course which allows for long-term transmission like HIV. But in the case of coronaviruses, peak titers occur very early, just around about the time that symptomatic disease has begun to be recognized. And then those titers stay high until around day 6 or so post-symptomatic disease, and then drop off significantly. So that transmission window is very short, but when you look at the disease, the severe disease manifestations typically occur after day 6 and progress for some time, several weeks thereafter. So that uncoupling of high rates of transcription from severe disease allows for transmission, allows for an effective transmission window, and consequently the evolution of increased virulence. I would also would predict that having a population with low level immunity would also select for viruses with increased virulence to circumvent that low level immunity. And so as we are in this expanding pandemic, there are waves of susceptible individuals, even those who've been exposed previously, that increased virulent viruses with variation in the spike to get around that immune response seems like a logical thing for the virus to do to promote itself in the population. At some point, I agree with you completely that a balance will be restored, and the most likely outcome of SARS-2 will be that it will cause disease much like the contemporary human coronaviruses. Thanks.
Lisa, did you have any follow-up comments on that?
No, I think Ralph answered very well. I think it is what it is. And I can just share a little bit of what we have been seeing here in Singapore. I mean, the recent weeks and months, we have also had cases of reinfection, and you are exactly right. This actually happened again in the foreign worker dormitories, and which some of them have already been previously confirmed to be positive, but they were on the very mild or even the asymptomatic side of it. So this is what we are actually seeing. And I think the worrying part about generating new variants of concern as this whole thing is still evolving, I think that is where everyone is watching. I think that's all I can add on. Yeah.
Thanks. Okay, so the next question externally for any of the panelists: what are your predictions for the durable immunity against SARS coronavirus 2 induced by either vaccine or natural infection? Is it possible a SARS coronavirus 2 variant will emerge that can induce a chronic infection that's not completely eliminated by host immune responses? So maybe we'll just go around the panel. If anyone has comments on that. Ralph, if you want to start.
Sure. We have studies in mice that show that the SARS coronavirus 2 mRNA or protein vaccines don't necessarily protect against SARS coronavirus 1 infection. And if you compare the change in neutralization response between the SARS 2 strains and the SARS 2003 strains, there's about a 30 to 40-fold reduction in nuke titers. So that's the advantage of looking at strains begins to give you an idea of where severe breakthrough might occur for some of these extremely potent and effective vaccines. So if variants of concern can evolve greater than 20% escape from existing immunity, then a broader swath of the vaccinated population is going to be very vulnerable.
Lisa, did you have any comments?
Well, what I would like to add to the one part of the question is about the protective response of natural infection and those from the vaccinated one. I think we are looking at potentially very different type of antibody epitopes as well. I think so. This is where it's. I think it leaves to be seen. You know how similar. What are the conserved epitopes and what are those that those that are generated from the vaccinated individuals. And I think we have a clear understanding of the whole repertoire of this. I think this is where we could potentially improve our understanding and generate better countermeasures. I think in developing new, more specific targeted monoclonal antibodies as well against the specific regions, as we know.
Yeah.
Linda, I'd also like to add, I think the vaccines are so powerful and elicit such good responses. It's so absolutely critical that we get the population, global population vaccinated. And it's in people's best interest because these variants of concern, if they truly are evolving increased virulence, the unexposed are at much higher risk for severe disease outcomes.
And I'll just follow up on the question. It appears that after natural infection, especially a moderate to severe infection, you have these persisting high antibody titers, including neutralizing antibodies that you don't see with an asymptomatic. So that may be important in terms of broadening a broader immune response than induced with just a spike, because you're going to bring all the coronavirus proteins into play, including N and E that also have T cell epitopes that's not in the vaccines. I think the question about chronic infection persistence is an interesting one. We have seen that some viruses, coronavirus, can persist in the tonsil. And there are reports now of persistence at least 4 months in asymptomatic patients in the gastrointestinal tract, either the antigen, in one case viral particles, or RNA. So how that contributes to the immunity, if that keeps the IgA system primed in terms of having a more persistent gastrointestinal infection, that's not known. But in that study too, they also suggested that people who had gastrointestinal infections alongside as part of the SARS coronavirus 2 infection also had less severe disease. So all of these are interesting things that need to be explored. I think we're just going to have a few minutes, like 30 seconds for each person to maybe comment about what do you think are the most important lessons learned from COVID-19 and pandemic preparedness, and what do you think is the future possibility for another coronavirus pandemic? So Ralph, you have about 30 seconds here.
Most important thing learned and future for the possibility of future coronavirus pandemics. So our lab has identified a large number of zoonotic bat and related coronavirus strains from 3 different subgenus that can infect human cells and replicate efficiently in primary human airway and intestinal cells. So I'm a firm believer that we could easily see another coronavirus pandemic, including within the cyber coronavirus group. And the reason I say that is that there are 2 contemporary human coronaviruses in 2A that co-circulate without any problem, and there's 2 contemporary human coronaviruses that circulate in 1B without any problem. And so there's plenty of antigenic space within the cyber coronavirus family to see another one come out of nature. What is the most important thing we've learned? We've learned that globally, we are not prepared for a pandemic. We learned the critical importance of collaboration and integration across the scientific community and the public health communities across the world. We've learned that as a species, we can respond in an extraordinarily positive fashion to get vaccines and drugs to people in an incredibly short period of time. And we've also learned that we can make tragic mistakes that have enhanced disease and resulted in increased death that shouldn't have happened. And so all the beauty and the plus and minuses of humans have been identified in this pandemic.
Okay, so with that, it looks like Ralph will get the final word. Sorry.
Ralph ran off the mouth too much, sorry.
And we've apparently run out of time. So I'd certainly like to thank the National Academy and all the panelists for their active participation and excellent pre-recorded talks that I hope everyone will have the opportunity to listen to. And with that, I thank you. And the panel is completed.
Thank you, Linda.
Thank you.
Thank you, Linda.
Thank you.
Hello, everybody. I'm Bill Press. I'm the Leslie Surgener Professor of Computer Science and Integrative Biology at the University of Texas at Austin. I'm also the elected treasurer of the National Academy of Sciences. This is the second panel presentation today. Our panel is about response to the pandemic. And we're very pleased to have here both scientists who helped respond and also Mayor Jenny Durkan, the mayor of Seattle, who will have many interesting things to say as someone actually making the decisions about what to do. Our format is similar to the last panel, except that we've asked these panelists not to show slides in their initial 5-minute presentations. We want to have more of a presentation with give and take, and of course we'll have the long discussion and question period afterwards. The other opening remark I should make is I'm going to keep my introductions of the speakers very, very brief, basically just saying what their current affiliations are. And the reason is if you're watching through the NAS website, you're just one click away from seeing their long and very interesting, in all cases, bios. In fact, if you find that link to click, my advice is to right-click it and open it in a new tab so that you can go back and forth between, so that you can listen to our discussion and at the same time, if you wish, multicast by reading their bios. So with that very brief introduction, the first speaker who will give a 5-minute kickoff is Dr. Nicole Lurie. Dr. Nicole Lurie, Nikki, is Strategic Advisor to the CEO and Response Lead for the Coalition for Epidemic Preparedness Innovations, CEPI. She's also a senior lecturer at the Harvard Medical School. I think more interesting and more relevant to our participation today is that during the Obama administration for 8 years, she was Assistant Secretary at Health and Human Services, responsible for preparedness and response. So she was not in government, of course, during the Trump administration, but she was in a very unique position to observe things on the national scene. And with that, Nikki, please.
Good. Thanks so much, Bill, and hi to all of you. Hope your Zoom fatigue isn't too great. So I'll just start by saying, as I think about strategic leadership here, the US had been planning for this kind of a pandemic for decades. We all know there was a lot in place. There was a playbook that should have enabled a different response for us and a different kind of strategic leadership. States and local governments have also planned for this for years. And at that level, it's stunning to see and to have seen such examples of exemplary strategic leadership, I think, in contrast to what we saw at the federal level. So the glass there is really very full, not even half full in my own mind. In my recorded talk, I outlined my perspective on a number of things that I think should have happened at a strategic or tactical level that did not. I wanted to touch on a few of those issues for the federal response here in the hope of stimulating, I think, some good conversation. So particularly from my perspective, having served as Assistant Secretary for Preparedness and Response, one of the responsibilities of government is really to prepare for the worst. You can always hope it doesn't happen. You can always scale back if it doesn't happen. But your job is to protect the American people from harms and to build resilience if the worst things happen. And so I think that there are a handful of areas that I want to highlight today where strategic leadership plays a really important role. The first of those, I think, is dealing with uncertainty and dealing with bad news. And as this pandemic unfolded and we had the early warnings a year ago, December, and then into early January, good strategic leadership would have done a couple of things. It would have done some scenario planning to look at all the possible ways this could evolve and plan for them. It would have gotten the public ready for those various kinds of contingencies, and in doing so would have served a terrific preparedness function for state and local government, for the healthcare system, for the private sector, for the public in general. And so thinking about how, as a strategic leader, one deals with uncertainty, and bad news is terribly important, and also how it is that you communicate that to the public. So understanding that that's the situation, also understanding the need is my second item to gain and retain public trust through a crisis is absolutely critical. And so that encompasses all of the communication strategies that I think we all know about at this point in terms of communicating clearly and consistently from the top, et cetera. And also, frankly, in the early stages, priming and educating networks of communicators around the country and around the world to carry those messages in a way that maintains confidence in government and in a really strategic way. A third strategic area that I would want to point to is eliminating single points of failure early. And I think we saw a terrific example of that in vaccine development when it ultimately got going, in that the federal government invested in multiple vaccine candidates because vaccine development is so risky. And it's been terrific to see so many of those candidates come forward and be successful. In contrast, we had a huge single point of failure when it came to diagnostics and diagnostic tests, with the failure of diagnostics development at the CDC and no real plan to go to the private sector and scale early. So eliminating those single points of failure as a strategic leader, terribly, terribly important. Thinking about equity from the very beginning is another important strategic choice. In every single disaster and crisis that we have ever had, and certainly any that I've dealt with, it is always underserved, disadvantaged populations of one kind or another, whether they're low income, low education, people of color, et cetera, people in harm's way in one way or another. So thinking about that from the beginning as a strategic choice, why they might be vulnerable, how to eliminate those vulnerabilities, not as an afterthought. From a perspective of all of us at NAS, setting a research agenda, particularly when you're dealing with the unknown and doing that early, terribly important. And the final strategic choice I want to talk about is thinking about the global implications of your actions early. I want to shift for a minute to asking a couple of questions. I think as I look at so many aspects of the failed federal response, one of the things I'm so struck with is asking myself, what kept really capable, competent senior officials from acting? And my observation has been that an atmosphere of fear and intimidation really permeated the science agencies and other parts of the administration, not just HHS, but others, leading to tremendous interference with science and lots of fears of people being able to stand up and speak out. So that once leads me to want to leave you then in my 5 minutes with a question for you, which is how, what as a scientific community is our strategic leadership opportunity and our obligation now? One of the things I feel strongly we need to do as a scientific community is think about how to protect the integrity of science and science agencies in the future and make it safe for scientists to do their job and speak out. But also to ask, what does preparedness for us as a scientific community look like, not tactically or operationally, but strategically? And what leadership choices do we all need to make now? Thank you.
Thank you very much. So I'm going to turn next to Mayor Jenny Durkin. Mayor Durkin is the 56th mayor of Seattle, and she became the city's first female mayor since the 1920s. I think there must be an interesting piece of history back then. And its second openly LGBT elected mayor. She took office in November 2017. We just heard from Dr. Lurie what might have happened but didn't at the national level. In fact, the national level proved, as we all know, to be entirely disorganized. So the baton fell really to our local, state, and local officials, of whom Mayor Durkin was really a shining example, and Seattle emerged as a great success story. So Mayor Durkin, what would you like to open with for us?
Thank you, Bill. It is really an honor to be here. And I want to thank the Academy for having me, and it's a very unique gathering of the Academy. This last year has been such a remarkable time, and perhaps one of the most sustained challenges that any of us have ever seen. For scientists, I know it was an unprecedented experience to actually live through a global pandemic and test on the ground all the theories that people have studied over 100 years and develop new theories to test those. I think as history is written, there will be many lessons. And the one I hope to contribute today is to better understand the necessary and important connections between public health officials, scientists and researchers, and elected political leaders. I think in the last year, we saw that when these links failed, our response to the pandemic was at its weakest. And when these links were the strongest, our response to the pandemic was in its strongest. As anathema as politics is to science, politics with a small p is critical in a successful response to a global pandemic. In the earliest days, the scientific public health system failed us. And that is, as we just heard, is mostly because of the testing. The failure of the testing regime and capabilities left all of us blind to the growing danger. And these early failures that left us blind really cast the die for the first weeks of the pandemic. But I think as the pandemic grew, we saw in graphic detail that the politics and not the science of the nation, the world, the region, or the cities, when it failed, we faltered. Seattle and the area was among the first in the country to experience the COVID-19 cases, hospitalizations, and deaths. We were one of the first cities to take bold action to curb the spread of the virus. And now, a year later, I believe we will be one of the first cities in the nation to reach a community immunity threshold by vaccinating enough of eligible individuals. Right now, we have the lowest number of cases and hospitalizations of any of the 30 major cities in America. But that is only because of the unified actions of our elected leaders, the actions of our residents, and because we have some of the foremost accomplished healthcare providers, labs, researchers, and scientists. But it's also because we elected leaders wed ourselves to those scientists and researchers, and we're willing to take bold actions at the beginning of the pandemic. We know that the only way to return to some semblance of normal is through vaccinations. And again, I think that we have to follow and listen to and promote the public health officials. Looking back 10, 20, or 50 years from now, I think we'll remember the sorrow and loss of this time. But I hope we collectively can also remember that when we did these works together, it was through bold action based on science that we actually were able to take actions that were unprecedented in this unprecedented time. Back then at the beginning of this whole crisis, we had no national playbook. And as we just heard, no national leadership. That left it to us, local officials, to actually write it as we want. If you'll remember the initial days, it almost seems rudimentary, where we're talking about non-pharmaceutical interventions, and started quickly moving the dial through all of our playbook. Here in Seattle, we had a tabletop exercise with my whole government when we learned about the epidemic in China, and we knew it would come here. But at that time, public health thought we had nothing to worry about because of the testing failures. And on February 28th, a Friday in the evening, I got the call saying we had our first deaths and hospitalizations in the area that have been attributed to COVID. We then government moved at unprecedented speed for government. But we were able to do so because we as elected leaders, the governor, the county executive, myself, and the leaders from the adjoining counties all got together in a room to decide how we could communicate to the public a unified message. By March 3rd, just days after we'd learned of the deaths, I was able to declare a public emergency. We were able to speak to the large employers within days of learning the information ourselves, and they were able to start moving their employees to work from home within the week. The data now clearly shows that was critical to us flattening the curve. And we in government had to do the same thing. We moved towards getting our employees working from home. And also realizing that we were going to have so many small businesses fail, our schools would close, that we had to turn the dial for government to provide services in ways we'd never done before. We started out turning that dial, but we had to do it very quickly. And it was only because of the scientists and researchers who, in the absence of the testing data that we would have needed to really see how big the epidemic was here, those researchers were able to guide us because we had what was called the Seattle flu study. I can't say enough about the work that they did. They went back and retested swabs and were able to determine that, yes, COVID had been here in our region for a while, and start doing some modeling based on genetic research they were doing of the virus itself. And it was one of the most sobering moments I've had in my whole life, and the most sobering moment perhaps in my time as mayor, because it was clear from the modeling that if we did not take bold action, we would have over 70,000 cases in a matter of weeks because the exponential growth was so great. You have to remember, governors, mayors, elected officials, we spend a lot of our time promoting the businesses in our community, drawing new jobs, having vibrant downtowns and restaurants and nightlife. We build that kind of urban place that is so important to us. And the only way that we could fight this virus was to shut that all down. And I think that that was one of the hardest decisions that any of us as elected officials, but because we were ground ourselves so early and so quickly in the public health data, in the pandemic research that we knew was there, and in what the scientists were telling us, it really was the only choice that we had. I think it shows that in those challenges, in those moments of crisis, that the World Health Organization was exactly right. That elected leaders needed to speak with one voice. So we made sure that the governor and the mayors and the executives were in a room. There was a lot of debate and disagreement on issues because we had no playbook and we were flying blind. But we had to trust the science, and we had to see, we saw in other places of the globe what was happening. And so we had to move quickly, and I think that that was the right thing to do. We have seen this same thing play out, and I think that we will have a fascinating way to look back at comparing those local jurisdictions that resisted the science with those that were wed to the science, and how their different cities both made it through this epidemic, but also how quickly they could recover. There will be 1 million case studies coming out of this time in history, but I think one is absolutely clear, is that we need both the political leadership and we need up-to-date scientific research capabilities, but we need it at a national and global level because the pandemic, being a global pandemic, we learned, I think, more quickly than in any other time how tied we are to other regions. So I'm very much looking forward to this panel and to some questions from the audience.
Thanks. Very, very interesting. And in fact, let me emphasize to the audience that if you go to the NAS webpage interface to this, I think that's not the same as if you're directly on the YouTube interface. You can ask questions right there and they'll be conveyed to us on the panel. Mayor Durkan described how she dealt with the rich scientific resources in her region. Let's go through the mirror now to a researcher on the other side of that equation. Professor Lauren Myers is the Cooley Centennial Professor at the University of Texas at Austin. She was the founding chair here of the Department of Statistics and Data Sciences. And she and her lab have pioneered the application of data-driven models and machine learning to improve the detection, surveillance, forecasting, and control of emerging viral threats. Lauren, what can you tell us?
Well, thanks so much. First, let me say what an honor it is for me to serve on a panel with these three brilliant and effective leaders who have really stood up as the adults in the room, guiding our cities and our country through this really traumatic period. So as Dr. Press said, I've been modeling epidemics for a long time, for decades. During crises like SARS and H1N1, Ebola, Zika, COVID, modelers like me have pursued answers to three basic questions. Where and how is the pathogen threat spreading today? Where will it be in the weeks and the months ahead? And how should we use costly measures and limited resources to slow spread to save lives? In other words, we model to understand the threat, to forecast the threat, and to mitigate the threat. And I'd like to highlight just three of the points that I made in my recorded remarks. First, we should have and could have had a data-driven framework for a coordinated and effective response to COVID across the United States at many different scales. As a proof of concept, after the 2009 H1N1 pandemic, my team worked closely with the state of Texas to build online decision support tools that would help with tabletop planning exercises, stockpiling of ventilators in preparation for future threats, pharmacy-based distributions of diagnostics and drugs, and vaccine distributions to ensure fairness, equity, effectiveness across the state. So these were challenges that we're facing today that we anticipated a decade ago. And these tools that we built would have potentially saved lives, yet they were shelved by the state agency as our memory and sense of urgency faded after 2009 and as the funding for these kinds of initiatives disappeared. So going forward, we must make sustainable investments in the science and the practice of pathogen preparedness at all different scales. The second point I'd like to make is in relationship to our ongoing modeling efforts that Dr. Press mentioned to support COVID responses in the city of Austin, Texas. The metropolitan area is about 2.2 million and growing very quickly. It's where I live. And Austin, like Seattle, is a great model of city leadership working with all stakeholders, as well as scientists, to make effective decisions on a day-by-day, week-by-week basis, and really working hard to cultivate community buy-in to those decisions. To my knowledge, Austin is one of the very few cities to clearly and consistently follow a color-coded COVID alert system for almost an entire year. This system was carefully designed by the leadership and the scientists to achieve very specific and intuitive public health and socioeconomic goals. It has also been instrumental in tackling two of the responsibilities that Dr. Lurie just mentioned, dealing with uncertainty and gaining and retaining public trust. So far, this alert system has been invaluable to achieving all of those goals in the city of Austin. Austin effectively socialized the policy, and I mean the leadership socialized the policy through press appearances, social media, and by developing and maintaining a simple public-facing dashboard that tracks the key data used to trigger changes in our color code alert stage. And they have thoughtfully guided the city every single time we've actually had to make a change and either relax measures or make them tighter. So my third and final point is really echoing what I heard the other speakers say in their recorded remarks. Dr. Rivers urged us to take to hopefully soon be able to take a period of reflection. And I just echo that this is an essential point in time for us to build a robust national resource in pandemic science, data, and modeling with direct bridges to policy and public health at all scales, from local to global. And I also echo Dr. Lurie, who urges us to develop and execute a real-time research agenda for dealing with the unknown. We cannot, again, fall into the trap of looking backwards and assuming that future pathogen threats are going to look like influenza or COVID or the viruses and pathogens that we're familiar with. It is imperative that we take a deliberate and scientific approach to overcoming our collective failure of imagination. And I look forward to the discussion. Thank you.
Thank you. Finally, let me turn to Professor Caitlin Rivers. Professor Rivers is a senior scholar at the Johns Hopkins Center for Health Security, and she's an assistant professor in the Department of Environmental Health and Engineering at Johns Hopkins at the Bloomberg School of Public Health there. Dr. Rivers' research focuses on improving public health preparedness and response, and she's been especially vocal in trying to look to what changes are needed in federal policy or the organization of federal agencies in light of the pandemic. Dr. Rivers?
Thanks. It's an honor to be here with you all today. I'm excited to join you. And we've heard from several of my colleagues the importance of bringing to bear science and particularly data and modeling to questions of outbreak preparedness and response. And so that is what I would like to highlight today. I think this idea of using public health data and analytics is really central to improving our public health capabilities. It's really what epidemiology is founded on, and I see so much room to grow yet in the field. The first overarching point I would like to make is that outbreaks happen way more often than people realize. It's true that COVID is a once in a century event, but it's about every 2 years that we have serious crises that rise to the level of threatening our national security. From SARS in 2003 to H1N1 in 2009, more recently COVID, Zika, Ebola, we face these major threats all of the time. And they're not just detrimental to human health and life, they also disrupt economic activity. And so there are many good reasons why we need to prioritize preparing for pandemics as one of our central capabilities. Now, what can we do? Often people think first of medical countermeasures, vaccines and drugs when they think of the kinds of things we should have on the shelf for crises. And it's true, I think vaccines are probably one of humanity's greatest achievements. And we should continue to bring to bear every resource that we can to expedite the development of vaccines and to develop therapeutics and to distribute them to every corner of the globe. But that cannot be the only tool in our toolkit. Even when we are fairly well positioned to be able to develop and deploy a vaccine, it can still take months and months and months to really bring that into the crisis. Ebola in 2014, for example, there were years of investments in R&D, and so there were several products nearly ready for clinical trials when the outbreak hit. And even still, it took most of a year to be able to begin those clinical trials. And during that year, it was through non-pharmaceutical interventions, the distancing, isolation, quarantine, contact tracing, that had already really brought the epidemic under control. We see with COVID too, it has been an absolute miracle year of science with the development of vaccines in under a year. But even still, it was a year, and a lot of unfortunately, many people lost their lives in that year. And so vaccines and drugs cannot be the only tool that we reach for. I think we need more innovation. I think epidemiology is built on data and data science, but we have not been prioritizing that for investment in the same way as we have medical countermeasures. And I think that for me is where data and modeling come in. There's so much we can do to bring modeling to bear to support public health response. We heard from Dr. Myers a few things that modeling can do to help support that decision-making cycle. Just to highlight some of those uses, forecasts or the expected future number of cases is one of the most common modeling products during an outbreak. And we can use those to anticipate what is going to happen in the future with the outbreak. Is it going to get worse? Is it going to get worse in one region relative to another? Where should we send interventions, and how should we plan our intervention resources, which are often quite limited when you're responding to an outbreak? Another common modeling product is testing various interventions to see what expected response, what expected outcome they will have on the outbreak, and to compare different interventions, which might be more effective and how should they be prioritized. I want to make the point too that there are a lot of parallels drawn between disease modeling and weather forecasting, and I'm a proponent of drawing those parallels because I think it helps people really understand the possible trajectory of the field. One thing I like to note, though, is that with weather forecasting, it was really not until the weather forecasts had come into a good cadence and were produced regularly and were accurate and reliable that we found uses for them. Originally, it was primarily about commercial activity, cargo ships, aviation, that were the primary consumers of forecasts. But once we got forecasting to the level that we enjoy today, we found all sorts of ways to use it and to understand it in our everyday lives, from planning to bring an umbrella to planning an activity at the park or indoors. And I think we can expect a similar trajectory of disease forecasting. We can imagine important uses for how we might be able to use those products now, but it's not until we really have them at hand that we will find the whole suite of uses. Right now in the US, there's a robust community of academics who volunteer their expertise in infectious disease modeling and analytics to support outbreak response. And I think it's such an incredible resource that we have. And it's really through the generosity and expertise of those academics that we have been able to power the US response. But I don't think we can carry on like that. That's not how we do national defense. That's not how we do weather forecasting. We have institutions that we fund and that we keep standing at the ready so that when a crisis hits, we are prepared to respond. And I think that's where we need to go with infectious disease modeling and analytics. And that's why I am so excited that the Biden-Harris administration announced on the first full day in office the intention to create a National Center for Epidemic Forecasting and Outbreak Analytics, which is very close to this idea of a National Weather Service, but for infectious disease forecasting. There's also a substantial amount of money in the American Rescue Plan to fund these activities. And so although the administration has their hands full with dealing with the crisis at hand, I'm very excited by their vision and their intention to push forward with creating this kind of institution. Happy to talk about that more in the Q&A, but just to close, after every crisis, and particularly every infectious disease emergency, there is a period of reflection where we try to understand what went wrong and what we need to do better next time. And that's because we know that there will be a next time, there always is, and it's true there will be another outbreak or another pandemic that we need to face. And I think that it is in our strategic national interest to be ready. And I think keeping outbreak forecasting and analytics at the ready is one of the key tools that we can have to be prepared for that next eventuality.
Thank you. I guess I'll exercise the moderator's prerogative and ask one or two initial questions. Mayor Durkin, we heard how you very quickly decided to follow the best scientific advice and were able to sort out what in your mind, in the mind of your administration, was the best scientific advice. We also heard from Dr. Myers a pretty good news story about how Austin, the city, did it. On the other hand, I know from having talked to Lauren previously, her story about how our state elected officials here in Texas were willing or not willing to accept scientific advice would be a very different story. So I'm interested from your point of view as someone right on the firing line. What was your previous relationship with scientists or understanding of your local institutions? What made you willing to jump in and follow the advice of a bunch of research scientists who happen to be in your area?
So that's a great question. I've had strong relationships with some of our great research labs. I had recently myself had cancer, and so had strong ties to both the Fred Hutch and others here, and the University of Washington as well. And I think someone asked before, I think there was a question in the chat is, what was it that made it different here? One of the key things was we as a region really believe in science. And so the leaders could lead, but it wasn't going to work unless people followed. And so I think not just having leaders listen to science, but having, a community in a region that really was steeped themselves in believing in learning and science really helped on that front. So I think having those relationships were important, but also making sure that as you progressed as a leader through all your decisions, that you were basing it on solid data and science. As Mike Bloomberg used to say, if you can't measure it, you can't manage it. And I think that that's steeped into mayors, particularly across not just America, but across the globe. And so having the relationships were key. I'll also say, and I hope we get to talk about it, is the relationships we're able to build across borders, so mayor to mayor, was critical, and across frontiers in other countries. I sit on the governing board of the C40 mayors, which is the climate international climate group. But we created a COVID recovery response that included mayors from each of the continents to be talking about how do we recover and how do we look out for each other. So I think that we need not just a national response, we need to have a more vibrant, connected international response.
Thanks. I think related to that, let me ask Dr. Lurie, you described what might have been at the national level if, for example, the Obama playbook that was well developed had been followed. At the same time, the actual authorities for public health are all state and local. The national role is almost completely advisory, at least when you go by what the statutes say. Is this an impediment for where we should be going in the future?
It's a great question. We live in a federalist society. I think we have seen that. And I think we've seen times like we did in COVID, where states and locals all went their own way or had to go their own way. But I think the reason for that was really absent federal leadership and guidance more than anything else. I don't know that federalizing all of public health is going to be either constitutional or going to be viable. But I do think, particularly as our data science has evolved and trade and travel and border crossing has evolved, thinking much more about how to defragment public health and what sorts of things need to happen at a federal level, particularly with just-in-time data sharing. It's really, really important. I think in past crises, we've seen lots of examples of where federal leadership providing guidance and information has taken us a really, really long way. And then it's often the publics and communities who say, well, why aren't you following the federal guidance, or why do things look so different here? So you want to have those degrees of freedom. We've also got a bit of a situation, I think, because so much state funding comes from public health funding comes from CDC, and yet the data sharing isn't terribly bi-directional right now. I think we have a situation where it's really hard for states to challenge CDC. And so I think really rethinking some of those relationships, super important. And also, as I said, some of the data sharing, it's crazy that data has to go from a local level to a state level, all get massaged and analyzed, and then go in a really fragmented way to a federal level. As, I think Dr. Myers said, or Dr. Rivers said, outbreaks happen all the time. We miss them all the time because we don't have good data systems and good communication that integrates across the federal, state, and local levels. So yeah, we can do much better, but we're going to have to do it in the context of the Constitution and federalism.
Yeah, you've raised data sharing, which is a very interesting question. I wonder, I bet everybody on the screen has some view on that. I wonder if some other panelists would like to chime in on how the data flows or doesn't flow now, and what should we do about it?
I'd like to see a big push towards making data, public health data in particular, open and available. Although I think greasing the wheels between the various levels of public health in terms of data sharing is critical. There are a lot of stakeholders who would like to make decisions based on that data who are not in that circle. There are often community groups, non-governmental organizations, elected leaders who are not connected to that system, who would benefit a lot from being able to better understand what's happening in their communities. And so I think the closer we can get to a kind of Census Bureau model where there are well-defined standards for how and when and at what level data should be released, the more stakeholders I think could really benefit from that data.
Mayor Durkin, were you concerned with data either flowing in or flowing out to help the national picture?
It was one of our biggest challenges at the beginning of COVID. I mean, we had, not only did we not have the testing data, but we saw quickly that, public health is a pretty narrow pipe and is pretty self-insulated. And so the data going in and out of that pipe was not being able to scale to what we needed. And it wasn't connected to things like our hospital system. The beginning, we did not know the capability of our hospital system in the region. How many beds were there? How many ventilators? What was their usage? And so we quickly built up working with the state, and we were lucky because we had Microsoft, Amazon, who lent us, engineers to build quickly a data system that would connect all of the hospitals so that we could see in real time where hospitals were stressed, what equipment they had, what PPE they had. I mean, and we were flying blind. We just knew we didn't have enough, but it was almost impossible to forecast how we're doing. So building that kind of capability, I think, is critical going forward. And people got to get out of the way. There's got to be less turf and more sharing. Because you just see people were so used to just sharing in their own space with their people, and they could always find rules and regulations and privacy things that would prevent us from really having a holistic look. And so I think we really have to learn those lessons and realize that when a pandemic hits, there's such a range of data you need immediately, from the failure of businesses to whether it's hitting our Black and communities of color harder. How do we get age groups, demographies, where parts of the state. So I think that we used kind of old world systems for a new world pandemic, and we've got to catch up.
I'd love to share Austin's experience with data. I think it's instructive as well. So early on, I think very much like Mayor Durkin was explaining, Austin had a lot of crappy data and was trying to figure out basically what should we be tracking, what should we, how do we know when to act basically. And one of the contributions the scientists made was to look at the data that were available sort of very rigorously and say, try to say which ones of these data sources is giving us a signal of where and how it's spreading in the city, and what's missing that we can very quickly try to access. And what came out of that was an analysis that suggested that the very best thing we could do on the fly was try to get all of our local hospital systems to report on a daily basis COVID-19 admissions. So how many people were newly admitted to the hospital? And that turned out to be a more reliable signal than cases, positivity, total number of hospitalizations, total ICUs, mortality, just for a variety of reasons. And so again, sort of a stroke of leadership, the mayor, the public health authority, the county judge, they collectively asked the hospitals to start providing this. And they've been providing it for almost a year now. Every day we get this feed, and that has really helped with situational awareness. And as we think about kind of building a national data resource, I think there's a few things we need to keep in mind. One is, besides all the data that has proved useful that we've had a hard time getting access to in COVID, I think it's really important that we approach it through socioeconomic, racial, ethnic equity lens. Even though we've seen a lot of data and more data, we still don't have a good handle on data that reflects disparities in real time and allows us to address those gaps. The other thing we need to think about is, in addition to having the data, we need to have the analytics and the translation so people understand what the data is telling them. Many people still think that when you move into these like upticks in the pandemic, that things are going to grow linearly. And they don't realize that they have to take action at the first hint of an uptick, right? It's just sort of this exponential growth. And so just having the basic analytics, the understanding, so that we can make sensible decisions when the data do become available.
So, you're reminding me of a couple of other really important points that I think would be good for us to talk about. One is, I've been really stunned when I look around the world at countries that have real organized health systems where everybody is in and the data's all in, how well they've been able to manage critical aspects of their response. And so if I look at a place like the UK or Israel or other places, they're able to use their health system to get pretty close to real-time data about the epidemiology around the country. That they've been able to use it in clinical trials to test all sorts of things. They've been able to use it in a diagnostic sense. They've been able to use it to execute a vaccine program. They've been able to use it rapidly to look at vaccine safety and real-world data and vaccine effectiveness. Our healthcare system, first of all, is so fragmented that we right now don't have the ability to do that. And because it and public health data usually travel on parallel tracks, never to meet, we don't have the ability, except in cities where there's really terrific leadership, like we've just heard about in Austin and Seattle, to make that happen on a local level. We're missing a national picture. And then if I go to the national level, to a place like CDC, and I think about all the people there in all the tools, we still have human beings analyzing most of this data over time, when right now everybody's drowning in data. And there are, new tools that have been developed through data analysts and data technology that could help a lot. And so save the humans for the real interpretation of data once a signal is detected, by a machine or machine learning or other kinds of ways. So it's also time to both integrate and modernize our systems.
Thanks. Let me change the subject a little bit. Caitlin described a proposal that she had published on previously in academic settings before the Biden administration picked it up, for something like a national weather service for epidemiology. We're coming out of, we hope, we're in a crisis where there hasn't been such a professional service within government. Instead, there's been a kind of a self-organizing volunteer effort of research scientists. So particularly looking at Dr. Rivers and Dr. Myers, is there a tension here? Can we have both a vibrant research establishment and an effective in-government establishment, or would they inevitably be in conflict?
Yeah, this idea has actually been percolating in the academic community for years. And so I think it's the academics themselves who see an enormous need for this. And I think that elected leaders and public health officials see the need too. What academics are really good at is advancing the state of the science, developing new methods, new data sources, really pushing the frontiers. What I think government is better at is doing the operations, really turning the gears, getting it into the hands of people who need to make decisions. So sitting with the decision maker as they go through the discussion. And so I think there's room for all of that expertise to be brought to bear. Right now, it's all being kind of shoehorned into academia. It's very difficult to build and fund an academic research program with operational support. And so I think that's where we need to grow what we're doing and move some of those functions into government, supported by academics who will really break that new ground for us.
Do other panelists agree with that?
I certainly agree with that. And I mean, I would say most of us got into this field because we want to do science that makes a difference. And the most frustrating thing is when we do good science, maybe it makes an impact on a local scale or not, and there's no way to sort of get it out to be so that it makes a difference, so that it, you know, advances public health and save lives on a national or global scale. And so I think a, you know, robust government infrastructure to support our kind of science, but then help us translate it into applications in the real world is absolutely vital going forward.
Yeah, I would echo that, and I think that it's, you know, tying some of the previous questions together. It's great to have CDC say here should be the standards, but if you don't have a way to get that down to the community level, then it's not going to work. But we got early in this pandemic, I think it was one of the greatest challenges was not just how do we solve the problem in front of us, which was just so significant and unprecedented, but what's coming next? You know, what is that forecast of what's coming next? You know, I was really fortunate. Not only did I have great local researchers here, but I was able to tap into some of the Obama people like Dylan George and Beth Cameron and Rebecca Katz and Jeremy Konindick. And I could ask them, okay, what's my next worry? Because I got to start worrying about that now. And so building that kind of vibrant infrastructure of having that modeling and having people that can say, not just here's the problem you have today, but here's the next 5 problems. So as you make this decision, be setting yourself up for the next 3, I think is so critical. And I think going back to that last statement is so true is we have to do it not just on a national level, we've got to have better connectivity again on a global level, because we have this pandemic because we didn't have that. And so I think whatever we model in America, we have to remember we have to stay connected to the rest of the globe.
And that also means not only human health data, but that whole field of animal health data, spillover, predictions, and analytic capability and modeling there, so that we really can do a much better job anticipating what's going to happen.
I'll just add one more point to that. It's, you know, the human, the animal, but what we've learned in COVID is that this is not just about the epidemiology and the biology. It's about the society. It's about educational data, housing data, you know, all of these sort of complex interdependencies. We need to be tracking them. We need to be mitigating the collateral harms that are happening throughout our society.
Totally.
Well, we have less than 2 minutes left, but there's a very interesting, almost philosophical question that has come to us from one of our viewers who says, what can an ordinary person do other than passively follow orders? Are ideas, imaginations, ambitions, and motivations of the 6 billion non-scientists all useless? Must this all be in government hands? Anybody want to have a stab at that in 1 minute?
I think that comes back to a point I made briefly in my remarks, that when we set out with weather forecasting, we had a fairly narrow idea of how we might use those forecasts. And it wasn't until forecasting became routine that we found a whole suite of uses for it. And like how we've been describing disease forecasts, originally weather forecasts were more at the corporate level or the federal level. And as they got better, it moved into the hands of individuals, and individuals became empowered to make decisions off of that data. And I would love to be able to grow into that with disease forecasting. So you might reach for a mask if the forecast in your area looks poor, or you might cancel your gathering. I could really see that becoming part of our everyday lives. I don't think we're there yet, but I think we could get there.
On the other end of that, I think this whole area of citizen science is really exploding. And it sort of links to this question about what can and do ordinary citizens do. Often they're the ones that get us the early signals about what's going on right now, right? And I think, again, bi-directionally with scientists, with data scientists, with others, asking the questions, having citizens around the world provide us eyes, ears, information, and then synthesizing it and feeding it back in the ways that Caitlin just talked about are just so critical.
And I think that the, ordinary residents and citizens, they don't just follow, they follow what they believe in. And so having the education and the information and transparency is so critical, because look, no city did well if people weren't willing to take the steps they needed to give up their job, to wear a mask, to keep physical distance, to not see the people they love. All of those were individual actions. And at the end of the day, it is only those individual actions that were our defense against this virus.
I think that's a great point to end on. We're just at the end of our time, so let me warmly thank all four of the panelists. I think we had a great discussion. Let me urge people who have watched the panel discussion to click through to watch the pre-recorded segments that each of the four panelists have done. I found those all fascinating. And with that, I guess we'll adjourn this panel. There's a short break, and then the third panel on the vaccination response, the vaccine development and vaccination response will be starting. Thank you all.
Thank you.
Hi, welcome to the third panel of the COVID-19 Symposium on Strategic Planning for Crisis. The third panel is dedicated to vaccines for COVID-19. And obviously, vaccines today are something that we have and we are deploying every day. But to go back probably 1 year ago, January, February 2020, when the SARS-CoV-2 virus started to basically be in China, get to Europe, other places, all people really started to ask when we can make a vaccine. And I was asked many times that question. Dr. Fauci was asked many times that question. And the answers of people like me that have been doing vaccines for many years was, well, usually to make a vaccine takes 10, 15, sometimes 20 years. This is a pandemic. We are going to go faster. We have new technologies, but minimum is going to take 18 years to have a vaccine. Maybe it's going to take 3 years. Things don't go well. So that's what what we were saying just a year ago, predicting when vaccines would be available. Well, reality bypassed every imagination that we had. Vaccines were ready in 10 months. And before the end of 2020, we started vaccinating. So how did that happen? Well, I think it happened for two reasons. One was new technologies. And the other one was an incredible investment from the public sector. About technologies, I wanted you to look at this first slide. Basically, I think what happened was the transition from what I call analogic vaccines to digital vaccines. So far, for under 50 years, we've been developing vaccines in a way that I call analogic vaccines, because to make vaccines, we needed to grow viruses, to grow bacteria, to grow parasites, grow them in fermenters, modify them, kill them, inject, or take portions of them, or modify them and inject. So in order to make vaccines, we really need to grow the pathogens. Well, that was I call analogic vaccines, because you need to have the material to make a vaccine. Well, this time we didn't need the virus. The virus was sequenced in China. The Chinese put the sequence on January 20, 2020 in the internet. And that sequence basically was teleported across the internet, every place in the world, and hundreds of labs the same day were able to download the sequence and make a synthetic gene and use that synthetic gene to make a vaccine without ever seeing the virus. So that's the difference. I mean, we've been going from analogic to digital because we didn't need the live virus in order to make a vaccine. Next slide shows what people did with basically with the synthetic gene. Once the people had the synthetic gene, they made 3 vaccines. One very fast by using the synthetic gene to make a synthetic vaccine made by synthetic messenger RNA. The other type of vaccine, the synthetic gene, was put into a viral vector, typical adenovirus, and that was grown, that was used as a vaccine. And the third type of vaccine at the bottom of the slide, basically, the synthetic gene was used to engineer a cell, mammalian cell or insect cell or a tobacco cell, and that engineered cell will make a protein that will be purified, combined with an antigen, and used to make a vaccine. So these are the 3 types of vaccines. We'll learn more about them later in this panel. So that's why we went very fast. The next thing is why we went so fast. And the next slide shows what happened. Basically, usually to make vaccines, we do things sequentially. First we do discovery, then to phase 1, to phase 2, then phase 3. And in between we pause, we look at the data, and that takes a long time. This time, and I mean, the pre-COVID vaccine development took between 10 and 20 years. This time, basically, everything was done in parallel. It was done in parallel because the governments, the public sector, the BARDA in particular, United States, spent more than 12 billion, which was given to companies basically so that they, to de-risk their investment, so they could do things in parallel. So discovery, phase 1, phase 2, and phase 3 were all done in parallel. And so things went extremely fast. And that's the reason we had the vaccine in 10 months. But beyond the technologies and the investment, we needed people. And the people that we have in this panel are the ones that basically actually made a lot of these things happen. It's with my pleasure that I'm going to introduce the people who are going to talk in this panel. The first one is Barney Graham. Barney is at the Vaccine Research Center at the NIH, has been there since a long time. And he has been working in many vaccines. He made a breakthrough in respiratory syncytial virus, and has been, he basically designed the vaccines or the construct of the spike protein that's been used by 90% of the vaccines have been developed. So Barney, it is your turn to tell us how you designed the vaccine for COVID-19.
Thank you, Reno. And it's my pleasure to be here with you today. I'm going to just briefly review what I talked about in the recorded message that's online. First, I just want to review the history of vaccines in one slide. And Reno already mentioned much of this, but the first two vaccines were made for viruses were made by growing virus in animals, either on the hide of an animal for smallpox or in the spinal cord of an animal for rabies. When Goodpasture discovered how to grow virus in eggs, that allowed yellow fever and flu to come along, and the next cluster was because Enders discovered cell culture and allowed large amounts of virus to be grown up for either live attenuated or whole inactivated. Then there was this era of molecular biology in which you could make reassortants or virus-like particles or recombinant proteins. And as Reno said, this took us up to bioreactors. And recently we had a gene-based antigen delivered by a VSV that was an approved vaccine now for Ebola. Now I think we're going into a new era of structure-based vaccine design, and we thought that RSV might have been the first one for that, but COVID snuck in between. And COVID is based on some of the principles that we learned during the RSV vaccine development, but it also took advantage of all these other new technologies that have come up really over the last 10 to 20 years. And I'm just going to show how this has been built, and we won't stay here very long, but a lot of this has been built on technologies that have been used to try to make an HIV vaccine over these last 40 years. And even though we haven't made an HIV vaccine, this technology that has derived from that effort is largely what's behind the current advances. We worked on RSV for a long time on the enhanced illness syndrome and created an immunological paradigm that would allow us to get through the regulatory pathway. There was many advances in human monoclonal antibody discovery, and we've had what I consider inadequate or failed attempts at pandemic threats when chikungunya came through, and MERS, and then Ebola in 2014 and Zika in 2016. That started us thinking a lot about prototype pathogen approaches that would be more comprehensive. And we started looking for ways to combine structure-based design or precision vaccine antigen design with rapid platform manufacturing. So our work with Moderna started in 2017, looking for prototypic approaches to pair myxoviruses with Nipah and MERS coronaviruses for the coronavirus family. And when this outbreak started, we already had a way of stabilizing the spike protein, delivering it by mRNA, and protecting animals from lethal challenge. So we were in a position not only with the technology, but with a roadmap and with a pre-established private-public partnership that was effective. So this is what put us in the position to go fast. And just now looking into the future briefly, these are the things I think are the things that we really need to understand to break through on vaccines like influenza, HIV, herpes viruses, etc. So, and hepatitis C in particular. So antigenic variation and understanding how viruses can maintain persistence and have avoidance strategies to be able to persist in the host. And a lot of the antigenic variation creates a problem of immunodominance or immune avoidance that we have to learn how to induce subdominant lineages of antibodies. And it continues to into inducing memory cells that can maintain over longer periods of time and in the right places, meaning if it's a respiratory disease, to have tissue resident cells in the lung. And I think we need to continue to improve our approach to engineering and delivery of vaccines, both for stability and cost, so that we can move these technology into developing country settings. So I'll stop there and keep going.
Well, thank you, Barney, and really thank you for this presentation, but thank you for really designing the vaccine for COVID-19. The next speaker is the one that basically is Larry Corey, is in Seattle, and it is the one basically has designed and coordinated all the major trials to make the vaccines available for people, basically coordinating all the trials in the US, making sure that we have efficacy, making sure that trials were done quickly, making sure that trials were done with all the vaccines were becoming available. Larry has been conducting trials in the field of HIV for a very long time, as far as I remember, and basically has been using his expertise and the networks that were set up to do vaccine trials, basically to do a remarkable, incredible work to make these vaccines available to people in such a short period of time. Larry.
Well, thank you, Reno. Again, it's a pleasure to be here, and I'm going to just show a couple slides here first that sort of summarize sort of where we are in the vaccine field. I would say that, just in reflection, that I think what the clinical trials program has shown is that when the scientific community, both academia, industry, and government, can get together and really work together, one can essentially get unprecedented speed and efficiency. Obviously, the large public investment made a difference. It allowed us to work with companies on an individual basis, but yet set up a structure that we could harmonize the endpoints. We could have common clinical trials organizations. We have common DSMB, really the unsung heroes of the program, and set up a sort of a system that allowed each trial or each vaccine, each platform to be individually evaluated. But yet when we come out of it, we know that there's some reasonable commonalities that we can get a sense of, sure, does it work, and how well does it work? It is this working together that I think we need to carry through in the future with respect to the pace and the cadence. And if you have that kind of pace and cadence, when you have success, how it can implement things. I mean, when we designed the trials, we thought from beginning to end it would take 6 or 7 months to do this. It actually ended up being 4.5 months. Now we had an amazing epidemic also that doesn't necessarily be a great thing, but the reality is that we finished these trials a couple months earlier than we even planned, with remarkable success. And we essentially made the trials large. We almost doubled the size over what we needed to get better safety data, get more speed. And I think the trials have been incredibly effective with respect to predicting real-world efficacy. The high efficacy demonstrated in the large clinical RNA vaccine trials appeared to be holding in the population rollout. It was largely based on the Israeli data, which shows high efficacy even with the B.1.1.7 variant, which is very gratifying. There's a large 300,000 person, 500,000 person trial of the J&J vaccine that is ongoing now in South Africa and the healthcare workers, and that also is starting to show real-world efficacy similar to what we saw in the clinical trial. So the recent data suggests the vaccines are having an effect on hospitalization and mortality. The more recent data say that some of these vaccines appear to have good activity against variants of concern. I think there's an emerging set of data that highly potent vaccines, RNA vaccines, the J&J vaccines, probably, and what appear to be the protein vaccines, at least the Novavax vaccines, all, not only are better than natural infection, but they actually have some widespread efficacy against variants. And I think rather than thinking about herd immunity, I really think that we should be talking about vaccine coverage or vaccine-induced immunity, because vaccine coverage contributes the best approach to also not only protect against disease, but likely reduce variant spread. I think the real issue is obviously vaccines in bottles don't abrogate epidemics. It's vaccination does. An extension of the best vaccines into the global population is what I think is our next need. Yes, it's a formidable task, but we have to create the infrastructure to do this. We need to recognize that giving vaccines that are only partially effective is really not going to be effective. I think the epidemics in Brazil and Chile and South Africa, in which we have neutralization resistant variants predominate, are showing that high natural immunity and the use of less potent vaccines is really not an effective approach. And we must bring a way of having more effective vaccines to the epidemics. I think we've learned enough so that we need to narrow down the choices that we are making available when we use vaccines on a large population basis. Your meningococcal vaccines, they were, used in lower and middle income countries. We didn't look for, quote, less effective, potentially cheaper alternatives. We want to give the best vaccines to the best people. I think one of the things we have to recognize is that immunosuppressed patients who have chronic COVID are the major source of what we call multi-mutational variants of concern. And we need to actually direct vaccinations to immunosuppressed persons. And the largest population of immunosuppressed persons globally are HIV-infected people, especially those untreated or having ART breakthroughs. That's 24 million people living with HIV in sub-Saharan Africa. In South Africa alone, there are 7.5 people living with HIV. They have a heroic antiretroviral effort, but there's still 200,000 new infections yearly. And we need to develop effective vaccination regimens for these people Whether that is J&J or RNA, or we need to think about making these vaccines more available to key countries where resistant variant epidemics are going to occur. Otherwise, we're going to have more resistance, and we're going to have the promulgation throughout the globe for this. And we need to think about creating manufacturing and supply chain and distribution of the highly potent vaccines globally. So I think that the neat thing is we've had a success story. We have effective vaccines. We know when we get good vaccine coverage, we can start making major advances on this epidemic with respect to hospitalization and death. But we certainly need to extend this globally, and that is going to require a concerted effort.
Well, thank you, Larry. Very compelling how vaccines were tested, how effective they are, but a big problem that still remains, which is basically how do we deploy these vaccines and produce these vaccines for the rest of the world. And as you said, designing vaccines by BARNI is great. Showing that they're effective and safe is fantastic. But unless we inject those vaccines into people, they are not effective. So the third talk about Amanda Horn from CDC is basically focused on taking the effective vaccines and making sure that we will be able to immunize people, which is an incredible task. Vaccines need to produce, need to be deployed. The organization needs to be incredible. The supply chain needs to be coordinated, and people need to be reached and injected. So, and everything has to be planned to be effective. And that's what Amanda has been, and her colleagues at CDC have been doing. So, Amanda, tell us how you did it.
Great. Thank you so much, Reno, for having me here today. And I'm going to wrap up these presentations with talking about not just the amazing work that got us these, right now, these 3 authorized vaccines in the United States, but actually the effort to get the country vaccinated. Excuse me. So we have delivered, we've administered over 215 million vaccines in the United States since the middle of December to this week. So this is over a period of about 20 weeks, which is just really a remarkable success. We've achieved 80% coverage in adults age 65 years and older, and that's at least with a single dose. And over 65% of adults age 65 years and older are completely vaccinated. And if you look at just adults over the age of 18, more than 50% of adults in the United States have initiated vaccination. And so we are getting to, we're getting closer to vaccine immunity, but we need to get even further. This slide demonstrates the incredible amount of work and changes that have been done over the last 20 weeks, primarily at the jurisdictional and the local public health level. What you see here in gray is most vaccines have been administered in local health clinics, in these vaccination sites that that local public health and state public health have set up. And then in yellow, what you can see are doses that have been delivered in pharmacies. And we have several federal programs where we've tried to augment the jurisdictional vaccination through places like federal retail pharmacy programs, and especially through HRSA, our federally qualified healthcare centers. Right now, we're also vaccinating in dialysis centers and in other places, really trying to augment in order to achieve vaccine equity, to make sure that no person is left behind and that we can really get vaccines into communities. I apologize. My dog has not barked this entire time until now. This slide shows how many vaccines we're giving per day. And what you can see is this has steadily increased over time. We've increased the number of channels that vaccines being delivered to. And up until last week, you could see we were steadily given, we were for the previous several weeks giving more than 3 million doses per week. Last week was the first week that we saw a decline in vaccination in terms of the total number of people who got vaccinated over the course of the week. And there is clearly the contributory factor of the pause in the J&J vaccine, which was called on by FDA and CDC last Tuesday. That pause was lifted yesterday, so we're excited about getting some of those doses that people had appointments for last week, getting vaccines in arms, either for J&J or for one of the mRNA vaccines this week and the upcoming week. But we also were seeing before that pause some leveling out of demand. And we really have to, this is a really critical time. I think the pause demonstrated that the United States is taking vaccine safety. Safety is the first priority. And we've delivered over 200 million doses of the mRNA vaccines with no safety signals detected. And when we quickly identified a safety signal with the J&J vaccine, FDA and CDC paused vaccination, and we spent 7 to 10 days making sure we understood everything we could about that safety signal, who was at risk, what was the degree of the risk, and looked at the risk-benefit comparison. And that's what was presented yesterday when the decision was made by the Advisory Committee for Immunization Practices to resume vaccination, making everyone aware that there is an increased risk for thrombosis with thrombocytopenia syndrome, which is a very rare thrombotic event that does appear to be associated with the adenovirus vector vaccine, J&J, as well as the AstraZeneca, but is still a very is occurring rarely. So in women about 18 to 49 years of age, it's occurring at about 1 per 125,000 women getting vaccinated. It is really critical as we move into this next phase of vaccination that we continue to build trust both in communities and across the United States, that we bring vaccine as close as possible to people. We want vaccines to be in every community. We know that healthcare providers play a critical role. So as we have more doses of vaccine, we want to make sure that primary care providers are vaccinating and that people can get vaccine through their healthcare providers, which is really some states are relying on their healthcare providers quite a bit, but that needs to be more generalized throughout the United States. And we're really working closely with community partners, community-based organizations, places like faith-based organizations, churches, local rec centers to sponsor vaccination clinics, either with pharmacies or with local health systems, to really make this a community effort to get the country vaccinated, community by community. And, we've made amazing progress. We're protecting older adults who are at most risk for hospitalizations and deaths. And we've seen hospitalizations and deaths decline in this older age group, but really to have the type of total impact to benefit from these vaccines, both the direct and indirect protection that happens when you achieve high coverage, we need to get the entire country vaccinated. And really the next big step in that will be in the coming weeks or months when we anticipate vaccines being available for younger children, going down first probably to age 12, and then hopefully over the coming months available for all children. And I'll close there. Thank you very much.
Well, thank you, Amanda. That's impressive what you have achieved and what you've been done. I think we got at the end of our 3 presentations from our 3 panelists, and now we will start the discussion. I'll probably start with the first question, which could be for probably Larry and maybe Amanda as well could talk about it. And because we heard an incredible story. The incredible story is that vaccines have been done well, they work well, they've been developed, shown they're efficacious, and deployed, and with the impressive figure of 250 million doses delivered in the United States. Incredible. So we have a beautiful, incredible story. At the same time, we know that this world, the world is basically 8 billion people. And Larry said, we need to vaccinate all of them. But we are not going to do that quickly. Some people say we don't have enough vaccines, and people say it's going to take up to 2023 before we can vaccinate all people globally. So the question is, we've done something which is remarkable, but we could do better if we. What would it take to do better? To be ready to make people globally faster.
Well, I guess I have to say that, this is where government and industry and the world need to get together. I sort of look at the issue of what we had with HIV and antiretroviral therapy as a model that we need to move to the model of sort of the concept of transferring the technology, tiered pricing, the development of the sort of distributive, sort of generic production of the high potency vaccines. So we get them distributed and made, not only for this epidemic, for the next epidemic. The ones that using the platforms that have staying power and that could actually be used over time to sort of force the investment. So I do think the concept of taking lower income countries and vaccinating only 20% of the persons is a flawed concept. I also think the concept that we should have 30 vaccines or 40 vaccines is also a flawed concept. I think that we need to concentrate our resources on highly active vaccines and figure out ways to get them distributed, manufactured and distributed as best we can. It's in our interest. I often say that, a generation is defined as Barney and you have said, by not only by inventing its technologies, but how it uses its technologies. Our humanity is defined by how we use our technologies. And I think this is really one epidemic that's causing death in everywhere. It has no borders, and it is really, and the economic benefit has been unbelievable. It's the pandemic of the last 100 years, maybe the pandemic almost forever, and it's absolute cost. And I think we need to bring resources to bear to globally to put this under control.
Yeah, I guess in the HIV that you mentioned was the PEPFAR, basically, project, where basically it was a huge investment from the United States that allowed the transfer of the technology to low-income countries that manufacture their own drugs. This is a scientific panel, but should we suggest something similar to the world?
I would. I say the road has been, we have a path. We created a path. Let's reuse
it has been successful. Yes.
Right. We have Gavi. We have global funds. We have the ability to do that. We need to build the production model, and yes, that will take some time. But we also have models in which we can increase production of our currently most potent vaccines and get into people's arms as much as we can. And the virus is telling us that we need to do this. Barney might reflect on that, but we're looking at these countries that have an enormous number of seropositives who are getting reinfected with still severe disease. So we have to hit them with vaccines that actually do work and prevent severe disease.
Absolutely. Well, thank you, Larry. Amanda, I don't know if you want to say something about this. Otherwise, I have a question, obviously, for you. But otherwise, I'm going to go to Barney. Barney, we've been talking about variants, about vaccines that are, they seem to work. Well, we know the present vaccine, some of them don't work or work much less against the variants. But some vaccines seem to work, but obviously second generation vaccines are already in the clinic. So hopefully we'll be able to control the variants which are out there. What's your feeling? We're going to have universal vaccines, at least for COVID-19. Is that possible? Or we'll have to basically every few years to adjust the vaccines again?
Yes, there's several parts to that question. I think as the epidemic or pandemic progresses over time, I'm hoping that it will become a seasonal disease. And then eventually it'll become a childhood disease, just like our other endemic coronaviruses. And so I think the types of vaccines, the need for vaccines is going to change over the next 3 to 20 years. The variants, I'm hoping, I don't know this, but I'm hoping that a lot of the initial change has been for transmission's sake and not for immune escape. And all the changes that have happened so far, even in the most difficult variants, like the one from South Africa or the one from India now with the 484 and 452 changes, I think that each of those amino acids can be tied to an improved infection or transmission, phenotype. So I'm hoping that this levels off and it's not like influenza, because for influenza, that's where you get into the antigenic variation, the immunodominance problem, some of the real difficult immunology that we really haven't solved yet. There are technologies, though, and several groups are now approaching this using nanoparticle display. You can make polyvalent or multivalent nanoparticles and cover a lot of different coronaviruses. I think it's going to be relatively easy to make beta coronavirus vaccines. Whether we can get all the alpha viruses and other coronaviruses, Delta virus hasn't infected us yet, but it might. And so I don't know if we'll get them all in one particle, but I think it's feasible now to make a pan coronavirus vaccine. But I think the real question goes back to what Larry was saying in your first question. We have to get the vaccines deployed so we can stop transmission, so we can stop the variants from emerging. If we can stop transmission, the variants will stop emerging. And so to me, that is the key step we have to take. And I loved your slide, your analog to digital slide, because this is the way I envision it. We skip a couple of generations of technologies for manufacturing and go straight to chemical synthesis and get this distributed around the world, just like we did for cell phones.
Well, thank you. And that was encouraging that you said we're going to have a universal vaccine. Actually, I believe it's possible too. I just need to continue working and watch. But as you said, I mean, if you want to stop this virus and stop the variants, we need to vaccinate people globally.
Yes.
Well, thank you. I think now we're going to go to some of the questions I had in mind mind for Amanda, but it's also coming from the audience. And obviously it's about safety and the clotting problem, the thrombolytic events. Amanda, you mentioned that already about the J&J, what the ACIP recommended yesterday, but there are still a lot of questions out there in the mind of people. And one specific question which is out there for the audience is whether the clotting problem is the same for J&J and AstraZeneca. And probably you want to comment a little bit broader about this.
Sure. And just before we go to that, I do want to say, we have to get vaccines deployed globally, but we also have to work with business, communities, and government to stop misinformation, because that is going to be, we can't get people vaccinated, even if they're available, if we don't figure out how to make sure that people really understand these vaccines and have the information they need to understand the risks and benefits. So the J&J, the thrombosis with thrombocytopenia, this signal that was detected in the J&J vaccine, in part, we were able to detect it so quickly. These cases are incredibly rare, so our vaccine safety signals would have detected it quickly, but we were super aware of it because there had been hundreds, or I don't remember the exact number, but dozens and now hundreds, I believe, of cases similar to this with the AstraZeneca vaccine, which is also an adenovirus vector vaccine, but they're different adenoviruses. So we don't totally understand why both of these vaccines are causing what is essentially being seen as the same syndrome. But it is likely, it is clearly very, the prevalence is incredibly low. It seems to be an immunologic autoimmune type of event. And these cases, if they're treated with IVIG, that is the treatment that these patients are recovering with. So essentially, the syndrome is being looked at similarly. We don't have the AstraZeneca vaccine in this country, so we haven't looked at the data from that vaccine as intensively at ACIP or at CDC in the same way that they have in Europe. But all of the results, and including the lab findings that are identifying the platelet factor 4 antibody and others are similar in both vaccines. I don't know if anybody else has anything more they want to add.
Well, I mean, the clinical syndrome, I think, is the same. These really very significant thrombotic events, often in the brain and the cavernous sinuses, but also elsewhere, associated with a high platelet factor 4, sort of a syndrome that's sort of like heparin-induced thrombotic thrombocytopenia, but of course heparin is not to be used. That actually makes the person's worse. So I think there are differences. There are more males associated with the AZ vaccine. I think it's been very hard to get numerator and denominator. There's been a lot more AZ vaccine, and there are well over 200 cases now in Europe. But the incidence, to be really accurate as to is one better than the other is really unclear at the moment, and I think we just need to get down to understanding that better.
Thank you. We got 5 minutes left, and we got 3 more from the audience. They're pretty simple questions, so please try to answer quickly so that we can do all of them. The first one is for Barney. And how does a single dose immunization with RNA vaccine, Moderna or Pfizer, compare to the J&J vaccine? Barney, are you
Yes, it depends. It depends on if you're measuring antibody responses or T-cell responses or clinical efficacy. there is single dose efficacy with the RNA vaccines. You can see a separation of the curves before the second dose, but it's clear that the second dose does bring almost everyone up to the same level of antibody, and so it makes it a much more uniform immunity across more people, and it's likely to be more durable. And so I think the second dose does help the uniformity, the magnitude, and the durability. And there is protection after a single dose of RNA. We don't have a way of comparing it for clinical outcome because they were tested as a 2-dose vaccine. We'll know more when J&J finishes their testing of the 2-dose J&J vaccine.
Thank you, Barney. Well, the second question is, can we discuss what's the most effective regimen to protect against variants, booster doses, new vaccines, whether the vaccine induced lower antibody titers in the elderly? Larry, probably that's for you.
I mean, I think real-world effectiveness is just starting to come out. In the really elderly, we don't have a huge experience. We didn't enroll that many people greater than 85 into the clinical trials. So we do know age has a factor here. So I think we just have to wait and see as to what will happen. I think booster doses are being looked at. The memory responses at 6 months look good. The data from Pfizer and some preliminary data that's not yet published from Moderna all suggest that the high efficacy in the 90s is holding for up to 6 months, and the antibody responses are holding up to 6 months. We know that there have been studies to show that there's a reasonable amount of B cells in memory. So there are things that of this wheat field that we have not cut down yet. We're learning as we go. We have very large cohorts now that will teach us as we go, and we have to set up monitoring systems to see if failure occurs, and if that failure occurs is because of waning immunity or variants. I think from my point of view, the Israeli data has shown 1 dose with Pfizer is like 40% effective. 2 doses in the real world is like 90% effective. Well, I want the 90% effective. I'm worried that the 1-dose vaccines in an area where there's variants are going to actually speed the emergence of escape-resistant variants. And I think that the regimens have to be what the clinical trials were based on, which is 2 doses for the RNA and going to be 2 doses for the protein, and we'll see how the rest of them work out.
Thank you, Larry. We have one last question, which I think probably is also probably for Amanda, which is basically, what is known about the long-term effects of vaccines? How do you convince people who are worried the possibility of serious long-term consequences? How can we convince them to be vaccinated?
So just to build off of what Larry was saying, we have many studies in progress to evaluate how long these vaccines will last for. And we have good data already showing that these vaccines there's good memory response at 6 months, but real world effectiveness, we have 4 to 5 months of data, and we are definitely seeing breakthrough cases, but we would anticipate seeing breakthrough cases. Even if a vaccine is 90% effective, when you have so much transmission, you're going to see cases in people who have been vaccinated. In terms of long-term safety, what I will say, and this is what I say to so many people, is we know that COVID causes, in many, many people, I think it's 16% of people right now, long-term symptoms and problems, neurologic, pulmonary, heart problems, and those symptoms can have serious impact on people's lives, and we've seen that. We have never really, there's no examples of vaccines ever causing problems that haven't been, safety signals that haven't been identified 6 to 8 weeks after vaccination. We vaccinated 200 million people with these mRNA vaccines. And those have included people from all walks of life. It's included pregnant women, it's included older adults, younger adults. And many of those individuals were vaccinated 4 to 5 months ago, and we've detected no signals. And we're following people. We have vSafe, which is a after vaccination checker, which continues to text people monthly to see if they've had any signs or symptoms of anything. And so I think the plausibility or the likelihood of there being a long-term adverse event associated with these vaccines is incredibly low. Nothing is, there's always some risk, but it's incredibly low. And we know that for people who do survive COVID, that there are long-term consequences in a high proportion of those people.
Well, thank you. I think we reached the end of the time allocated for this panel. I really like to thank Barney, Larry, and Amanda. I think they've done a fantastic job, and it's really a privilege to have in a panel the 3 people that really made all these things happen. I really want to thank you. Also want to thank the audience and the National Academy of Science allowed us to have this conversation. I think this is the end of this panel. We're going to go on with the 4th panel in 10 minutes. Thank you very much to everybody.
Thank you.
Pleasure.
And thank you to the audience.
Thank you, Reno.
Thanks for the nice moderation, Reno. Thank you.
Hello, everyone. I'm Marcia McNutt, and welcome back to today's symposium on strategic planning for crises, with particular focus on COVID-19. I'm Marcia McNutt, president of the NAS, and it's my privilege to moderate this final panel on the use of scenario development in order to be better prepared for an uncertain future with COVID. I'd like to turn now to my panelists for them to each introduce themselves before I give some opening remarks about the topic of this panel. So Karen, why don't you begin?
Hi, everyone. My name is Karen Dynan. I am an economist by training, and I am currently a professor of the practice at the Harvard University Economics Department and at the Harvard Kennedy School. And I spent most of my career before coming to Harvard in policy jobs in Washington, D.C., most recently as the Assistant Secretary for Economic Policy at the U.S. Treasury Department.
Okay, Gary, you're up.
Hello, everyone. My name is Gary Mackless. I'm University Professor of Environmental Sustainability at Clemson University. And prior to that, I served both terms in the Obama administration as science advisor to the director of the National Park Service and co-led the development of the strategic science group that collected for Department of Interior leadership interdisciplinary scenarios related to Deepwater Horizon and Hurricane Sandy.
Thanks, Gary. Okay, John, let's go to you.
Good afternoon. I'm John Lumpkin. I'm an emergency physician by training and formerly the director of the Illinois Department of Public Health. Currently, I am vice president for Drivers of Health Strategy at Blue Cross of North Carolina, and I also am president of the Blue Cross North Carolina Foundation.
Thank you, John. Okay, Janet, to you.
Good afternoon, everybody. My name's Janet Napolitano. I've had a long and varied career in public service and now in academia. I began as the U.S. Attorney and Attorney General for Arizona. I was the two-term governor of Arizona. I was then appointed by President Obama to serve as his Secretary of Homeland Security, which I did for about 5 years. And then I joined the University of California as the system-wide president of the university. And beginning this past fall, I joined the faculty at the Goldman School of Public Policy at UC Berkeley, where I'm currently serving as a professor.
All right. Thank you, panelists, for those introductions. So I was listening to the previous panels and I realized that the one that Bill Press chaired set us up perfectly for this conversation. I took notes here that Mayor Durkan said, while everyone is worrying about what's happening now, no one is worrying about what are the next 5 problems that we need to deal with in terms of this pandemic. And that reminded me of a very famous quote from the physicist Niels Bohr, who said, "Prediction is very difficult, especially about the future." So when you face an uncertain future with a pandemic, what tools are available to look into the future to see what you might have to deal with and how you can be resilient to it, but and do it in such a way that you aren't constantly fighting the last battle, that you aren't relitigating the 1918 flu or Ebola, because each pandemic will be different and you have to be prepared for what could come down the road in the future. Well, in Bill's panel, Nicole Lurie already dropped the idea of the importance of scenario planning. Scenario planning so that you can be prepared for that uncertain future and you can identify single points of failure that would actually cause huge disruptions, such as she said, the single point of failure with the US testing protocol. So if I could have my first slide, please. What I've done here is a quick 1-slide summary of Martin Sepulveda's excellent presentation, which was available online. But since I know not all of you are overachievers and looked at all the videos before the panels, I'm going to do a quick summary of it. So basically, this chart shows the basic principle of scenario planning. That scenario planning is trying to understand what possible futures do we need to anticipate when the future is dictated by forces beyond our control. So in this case, I've chosen two axes that are outside of our control as scientists or perhaps even as individual members of society, but could lead to very different futures. On one axis, I put, what is the virus doing? At the top of that, I say it's a one and done. It's a simple virus. We develop a vaccine. The vaccine works, and that's it. But on the other end of that arrow is the possibility that the virus could be constantly mutating to vaccine-resistant forms. We heard in the last panel about the fear that it becomes like the flu, constantly changing, coming back every year, and in a form that is not addressed by last year's vaccine. And then the horizontal axis I have here is, how does society respond to complex challenges? Is society very proactive, masking, social distancing, washing hands and making sure that everyone gets vaccinated? Or is it reactive to challenges, that people refuse to take precautions until they are personally affected by it? So if you look at the spaces that are divided by these by these two lines, we have four quadrants. Scenario 1 and scenario 4 are quadrants where the virus is actually acting in a very easy way, but society could either be proactive or reactive. The lower two quadrants, scenario 2 and scenario 3, are a very difficult virus to deal with, but society either could be proactive or reactive. And of course, we could put different countries around the globe in either scenario 1 and scenario 2, or in scenario 3 and scenario 4, depending on how the people of that nation respond. So once these scenarios are defined, then the next thing that happens is different teams of researchers who are broadly constituted across the life, social, policy worlds, will indicate what would be resilient actions we could take right now if we found ourselves either in scenario 3 or scenario 4 or scenario 2, etc. What are the resilient things we should be doing now? And what usually comes out of this scenario planning is that interventions that are useful in one quadrant end up popping up in other quadrants. So those are the no regrets actions that we should be taking now in the face of an uncertain future to make sure that we don't have a cascade of negative consequences. So with that introduction, I'd like to turn first to Karen, because Karen has led the first scenario that the academies are developing on rental evictions. So Karen, the floor is yours. Tell us a little bit about this work.
Thanks very much, Marcia. So I'm going to start with a few words about the importance of the work of this strategy group, the topic that we are addressing. So we've been grappling with the task of producing a set of actionable recommendations that will reduce and mitigate the wave of rental evictions that may result from the COVID-19 pandemic. So we've seen considerable job loss, and we've seen much income replaced by government interventions as yet. But more importantly, rental evictions have not been a problem as yet because the federal government and many states and localities have had moratoriums on rental evictions. But the federal moratorium on evictions will be lifted at the end of June, And estimates suggest that notwithstanding all the government support that we have seen so far, more than 10 million renters are currently late on payments, with the typical delinquent renter owing something like $6,000 in back rent. Okay, so hence the fear that this is going to result in a wave of evictions from rental properties. Now, the good news has been that recent fiscal packages have passed a substantial amount of funding that states can use to help distressed renters. However, particularly given what's at stake, we cannot take it for granted that this money will solve the problem. The federal government needs to get the money to states, and then the states need to get money to the right people. And as we are trying to help distressed homeowners in a great recession, people who are having problems with their mortgages, states have very different capacity in terms of their ability to reach the right people at the right time. So this is all what the group is working on to put together this package of recommendations. And just to build on what Marcia was saying, setting up this panel, scenario planning really was the right tool to be using for this exercise. I should say I spent much of my career in organizations that at the time were making policies around a single baseline forecast, which is an approach that serves us well in normal times. But that approach just isn't suited for times when there are considerable uncertainties about where we're headed, such that the best package of policy is one that is going to be robust to a range of possible scenarios. So our group considered 4 scenarios that reflected different mixes of outcomes in terms of how the virus plays out and in terms of what other disruptors might arise and how they would be responded to by society and the government. And we went all the way from a world where there was an effective response to the virus and external disruptors were not a big issue to a world where policy responses were fragmented and there's an onslaught of external disruptors. So these scenarios were just meant to be examples to focus the group. It was a tool, as Marcia described. We did not literally craft recommendations for each scenario, but it proved to be a useful tool for getting the group to stretch their thinking in terms of the right, robust package of actions to be taken. Now, unfortunately, I cannot tell you the results of the report since it is still in review, but I do want to just close out by offering 3 of the lessons that we learned with the first strategy group. And the first is that it turned out to be very important to have a large strategy group that spanned many disciplines and mixed both academic experts with practitioners. So the rental evictions issue it's very complicated. It has numerous stakeholders, and there's going to need to be quick policy turnaround. So it was just essential to have people with knowledge about institutions and the real world challenges that renters, landlords, and communities face. We also heard from people with lived experiences, who were facing eviction from their rental properties, on how they saw the causes and the solutions to housing stability and the costs of housing stability. So that's the first lesson. The second lesson is that it's actually been really challenging to adapt processes such that we move with the necessary speed while still preserving the key hallmarks of academy work. So, we're looking for objective, independent, evidence-based analysis. We want to have a rigorous review process. We want to have actionable recommendations. And as we were kind of working with the group, we learned a lot about what worked and what didn't, and we'll be building that into processes for future strategy groups. The third lesson builds off the second, which is the importance of managing the scope of the process. In our case, we knew the primary task was to address the acute wave of housing instability that may be arising from the COVID pandemic. But the fact is, we know that there is way too much housing instability for renters on a longer term basis, and that those longer term problems exacerbate the shorter term problems. So a key lesson we learned was figuring out where to draw the line is going to be essential to ensuring that turnaround is sufficiently rapid. So those are my 3 lessons. And I guess the last thing I just want to do is to thank Marcia for her leadership, to thank everyone for their huge efforts. Our strategy group members spent many hours helping us. And I think that the Academy's staff spent what you literally might call countless hours pulling this all together. So I'll leave it there.
All right. Thank you, Karen. That was a wonderful summary. I think it's going to be interesting to go now to Gary, because your scenario is something that I think everyone can recognize as an important one, because all of us either know someone who's facing housing insecurity, or we hear about it on the news, etc. But now Gary is planning a second scenario, and this is one on ecosystem services. And I think Gary's situation is one that maybe is not first and foremost on the minds of Americans, but perhaps it should be. So Gary, tell us what you're thinking.
Thank you, Marcia. It is plausible and sobering that the economic and social disruptions caused by the global COVID-19 pandemic are the largest and most systemic since World War II. There was the ubiquitous blackout curtains, and now the mask. There was the rationing card for sugar and meat and petrol, now matched by the grocery sign that says limit 3 per customer. And then there is the immense death of civilians. Now, I do not mean that they are comparable, but they are equally cruel. This very morning, the Johns Hopkins Coronavirus Resource Center reported that we have crossed over past 3 million global dead. Here's a standard definition of ecosystem services, which forms the basis of the scenario work we're planning. It includes a scenario of how the COVID-19 disruptions may impact ecosystem services over the midterm, 5 to 10 years, and what the implications are for sustainability and resilience using the standard definition. There are some evidence-based clues. City lockdowns here, Rome, Madrid, New York, and Delhi, have led to significant declines in nitrogen dioxide concentrations. In India, the particulate matter and soot declined 25 to 30%. The result was a decline in soot and particulate deposits on the snows of the Himalayas that led to an increase in albedo, increasing the reflectivity of solar radiation, and markedly slowing snowmelt, altering the timing and levels of water supply for millions of people downstream. The volume of PPE waste created by the COVID-19 precautions overwhelmed waste management capacities in many countries and has led to significant shoreline and marine pollution. Oceanasia estimates an astounding 1.56 billion face masks, that's 4 to 6 million metric tons, entered the oceans in 2020 alone. Some of the effects of the COVID-19 disruptions have actually been positive to ecosystem services, though the trends are unclear. In Phuket, Thailand, beach closures and the abrupt disappearance of tourists have seen a thriving hatchling season of leatherback turtles, these are in the Muang Sanctuary in Phuket, that have returned to the tourist abandoned beaches. Yet in parts of West Africa, the absence of safaris and the loss of income has reduced wildlife patrols and led to increased poaching. Available data suggest a complex and extremely fluid set of drivers impacting ecosystem services. And that should not surprise us, given, as Marcia and others have said, the disruptions themselves remain complex and fluid. Hence, there are some significant challenges to the ecosystem services scenario that we need to pay attention to. We're going to have to distinguish between what are temporary disruptions and changes, what are transitional to a new future, and what are deeply structural changes. We need to identify causal pathways to avoid spurious correlations, and that means being extremely careful on the chain of consequences linking these changes to COVID-19 disruptions. To have an evidence-based scenario requires high-quality data at the appropriate scales on which you are examining ecosystems. And we need to identify key uncertainties all along the chain of consequences and the uncertainties underlying the assumptions that the scenario must necessarily make. And all of that to describe potential policy and management responses. These responses are critical, for we depend on ecosystem services for whatever resilience our species may have. And the fate of ecosystems is our own. Thanks.
Gary, that was wonderful. Your opening about the comparison of COVID to World War II was actually very apt. Earlier today, I showed a slide from The Guardian that shows that the annual death toll from COVID-19 has surpassed World War I, World War II, and the Civil War in terms of its impact in the US. So, John, I'd like to turn to you next. You are responsible for a massive healthcare organization. What are your views on these scenario developments, and how do you think they can help the American healthcare industry?
Great. Thank you, Marcia. And, this has been a trying time for healthcare. We've seen hospitals and healthcare providers who've been overwhelmed by wave after wave of this pandemic. And at the same time, during the ebbs, they've had to play catch up with many of the preventive services that are required to mitigate the impact of delayed service and foregone analysis and identification of cancer and other diseases. The public health system has been overwhelmed by COVID. First, in understanding the epidemiology. And this is a system that was devastated in the economic downturn of 2008 and 2010 through 2008 and 2009, when over 60,000 local health department employees were let go as part of that economic downturn. And that workforce was never replaced. The challenge of getting out the messages about what are the public health steps in the midst of a politically charged environment. And then setting up one of the largest vaccination campaigns in our nation history. I know from having been on those front lines that too often we look down, not down the road, but to the next patient, the next crest of the pandemic. Yet we don't have time to look at what the trends will be. What will be the impact 12 months, 18 months down the road? We just heard from Karen about the impact of what will be happening related to evictions. And at the same time, evictions are associated with increased use of emergency department and other healthcare services. We heard from Gary about the impact upon the environmental services, the eco services, and we know that that has a direct impact on health such as asthma and other diseases, and that they disproportionately impact communities of color. What has been outstanding about this process is that when we understand that health is about more than healthcare, and the decisions that need to be made in the healthcare setting and the public healthcare setting are so much determined by factors that are beyond the control of those same systems. That is exactly the kind of setting, as Marcia talked about, that are important to consider scenario planning. And what the National Academies did in establishing this process was create a parallel process, a process where each of the academies were setting up committees to address the immediate impact of COVID. What do we need to do to think about with vaccinations? What should we be thinking about treatment? What treatment works, what doesn't? At the same time, this Response and Resilient Recovery Strategic Science Initiative was setting aside effort within the academies to think down that 12 to 18-month path. And I think that we can demonstrate that this will have a positive impact. In addition, once the reports are prepared, it will be critical for all the academies to look at those reports and the scenarios and the trends to identify the impact. It will have an impact upon where I work at Blue Cross of North Carolina. If we can expect increased evictions, we have to think about the non-clinical drivers of health, those social determinants of health that will be impacted by each one of these scenarios. So it's been a pleasure to work with such a talented crew, because the multidisciplinary approach that was taken in this panel enabled different perspectives, from the ecology to the environment to the legal and regulatory, as well as the housing and economic aspect. And that is always a critical, important component of looking and trying to peer into not the future, but the potential futures, and with the scenario process, determine what steps may best prepare us and position us to be able to protect the health and well-being of everyone in our nation.
Wonderful, John. Thank you so much. And now I'd like to turn from the actual focus, maybe just on this current crisis, to Janet, who has certainly been on the front lines of many crises before this, and get some higher level view of this scenario resilient recovery framework, and what your thoughts are for how we can benefit from using it.
Thanks, Marcia, and thanks to Karen and Gary and John. It's been such a pleasure serving on the steering committee with you for the scenario planning initiative. And I hearken back to my time as a governor and as Secretary of Homeland Security. When you're dealing with a crisis and you are in a leadership position, you are at some point overwhelmed with coping with the immediate impacts. And you really, you kind of know in the back of your head that there's a future out there, but you don't really have oftentimes the capacity to even think about it. You know, the topic of housing and evictions. Housing is one of the most complicated public policy areas that we have in our country, overlaying jurisdictions, regulatory and legal requirements, and by the way, a direct relationship to the homelessness issue that is an impact of eviction. Ecosystem management, so very, very important. My first exposure to this kind of scenario planning was in managing the Deepwater Horizon oil spill in the Gulf of Mexico, working with the Department of Interior, the EPA, the White House, the Coast Guard, and then state and local jurisdictions, as well as with BP to identify how to put a stop to that gush of oil at the bottom of the Gulf. and also to deal with all of the impacts experienced in the coastal states that were in the immediate area of the spill. Here's the thing. This kind of a process conducted by folks, experts from academia, but also practitioners who can have a robust engagement that can help think through and plan ahead and raise, like, you need to think about this, and you need to think about this, and you need to think about that. And here are interventions that you can put into place that will help mitigate future harm. And that will help refocus leadership, not just on dealing with the crisis of the day, but mitigating the future crises that can occur unless you do something like scenario planning. So to have the Academy put its weight and its processes behind this sort of exercise is a wonderful contribution to future crisis response and planning in the United States. I dare say it would have been a real help to me as a governor dealing with, I was in Arizona, we had a migrant crisis at the border, we had major forest fires, we were accepting and accepted refugees from Katrina who would arrive in Arizona almost unannounced on a plane and get disloaded at the airport. So to have at my fingertips the kind of scenario planning, both the substance of it and the process of it, would have been quite advantageous. And certainly as Secretary of Homeland Security, which is a crisis job, leaning forward and thinking toward the future and not just being reactive, but proactive, as Marcia's chart showed, would make for a much stronger Homeland Security enterprise for the nation. So I think that as these scenarios get completed, as the reports get produced, we need to really spend some time making sure that the public leadership of the country, the mayor of Seattle, to the Secretary of Homeland Security and everybody in between has access to them.
Wonderful, Janet. Thank you very much. So now we're going to launch into some Q&A. So if anyone out there wants to send some questions, please send them in. Has Martine Sepulveda joined us? Martine is our fearless leader in this Response and Resilient Recovery Strategic Science Initiative at the Academy. I hope you saw his video that he pre-recorded. But in any case, Martine was hoping to join us for the Q&A. And so please let him in. But in the meantime, I have a question. This is really probably mostly directed at Gary and John, but if anyone else wants to weigh in, that would be great. Gary and John, you both talked about situations where people are worried about the near term and maybe not focusing enough on longer term cascading consequences. In this particular case of COVID-19, and let's take Gary's picture of the plastic that and all the trash that's going into the ocean from personal protective equipment. How do we weigh the cost benefit of the near-term reduction in infections by encouraging the use of PPE versus the longer-term problems that massive use of PPE and then poor disposal has for the ecosystem services, without which, of course, this planet becomes uninhabitable. So do either of you want to attack that one? Gary, you look like you're ready to jump up.
And this borrows from Janet's wisdom about thinking 5 steps ahead. The very moment we knew we needed to ubiquitously wear masks, the waste management systems of the United States should have been armed with scenarios. What's going to happen downstream? And the design and creation of these masks should have been done in such a way that they minimize the long-term pollutants, the microplastics, for example, and the waste management systems should have been given a burst of engineering, a burst of science, a burst of support to get ready for what was obviously, and I mean it metaphorically, downstream. And so I think the 5 steps ahead, the potential unintended consequences, and the quick movement to realize if we have a health problem, we also have a waste management problem.
Excellent. I agree. John, do you want to add anything more before I go to audience questions?
Absolutely. And, you know, it's the old adage, when is the best time to plant a tree? 20 years ago? When's the second best time? Today. We knew for 100 years that we would have another pandemic, but we always thought it was going to happen on someone else's watch. And I think one of the greatest lessons out of this pandemic is that all of us, including the academies, should actively engage in scenario planning. So we're asking the questions, and then what? And then what do we do? And then what should we do? You know, Gary's example of PPE and what will happen with that is a perfect example. But another one is, you know, hospitals and other healthcare providers have been actively engaged in just-in-time inventory management. And we never ask the question, what will happen if there's a pandemic? We need to start asking those questions more. And then secondarily, when a major event occurs, a pandemic, a major drought, the process of setting up a scenario planning process at the same time as the response is critical. To bifurcate those two activities so we're not only thinking about what to do next, but what to do after that, after that, after that.
Good, John. Yes, so the framework that we've used at the Academy is we wall off the actionable science delivery, which is the just-in-time advice on wearing masks and crisis standards of care and the value of social distancing. We wall that off from the strategic to make sure that neither one is getting shortcut. And I love your analogy of people always assuming it will happen sometime, but not on their watch. I think anyone involved in crisis management or the use of science in crisis, you should all have something on your desk that says it is your watch. And so that we're always prepared for that. One other thing I'll mention is I saw this fabulous little sign at my grandchild's preschool that said, treat your mask like you do your underwear. And all the kids had cloth masks and they were being encouraged to make sure they got washed every day and that you had a clean mask when you went to school. And I thought, wow, you know, the preschoolers get this. Why don't the rest of us understand that cloth reusable masks washed every day are a great solution? So let me go to some of the questions that are coming in. I've got one here. One of our viewers is asking, how can the results of scenario planning become a fixed part of the federal government's activities so that it can't be canceled by a future leader? Maybe Janet, that one's for you.
Yeah, that sounds like a Janet question. That's tough because, under our system, things that are done by the executive branch can be undone by a succeeding executive branch. If things are actually put in formal regulation or statute, it's much more difficult to undo them. If something is an actual regulation, to undo that regulation, you actually have to do another whole notice and comment period and the like. And of course, doing anything through Congress is extraordinarily difficult. And so, I think the easy answer would be if scenario planning leads to concrete recommendations for mitigation, et cetera, is to attempt to put them into regulation, if not statute. But I would also say that we need to imbue our civil servants, the career civil servants, with the values inherent in scenario planning, because career civil servants are a wonderful resource in this country, and they don't change from administration to administration. And so having a dedicated kind of reach out into the civil servant community, career civil servant community, would be another recommendation.
I just echo that, having spent so many years at different government agencies, I think you're just right about that, Janet. I mean, these government agencies are staffed up with technocrats who are natural candidates to be doing this analysis, and we need like a cultural change that just just changes the way they do normal business and how they think about it.
Yeah, good point. John, did you have something you wanted to add to that?
I would just add to that the role of science is so critical, and particularly science out in the public. And that is why the role of the National Academies is critical as a process and a place where that scientific discussion can occur, which sometimes can be challenging within agencies, but it creates a check and balance, and I think a very important one.
Yeah, good. And Gary, you've got something. Yeah.
If Katrina and Hurricane Maria taught us anything, it is not to rely on the federal government to be the source of restoration and response. And I think governments at the different levels, when county governments start to do scenario planning, when independent universities do scenario planning for the local regions that they have built trust and experience on, when all of that starts to happen and we don't just focus on the federal government, I believe we'll have a more resilient scenario ability to respond to the next crises.
Yeah, I think that's a good point, Gary. I once was part of a panel that was at an organization in which there are leaders for county and local government were all participating in this meeting, and it was an association that they all belonged to. And we were talking about the science capacity. And of course, the federal government has immense science capacity because there are scientists embedded in almost nearly all of the agencies. But when you get to the city and county level, they do not employ reams of scientists to do the kind of advising they need. So we discussed on this panel, what are options to get better locally valid science into the planning for states and counties. And we concluded, just as you did, Gary, that there are so many community colleges and local universities that do have capacity, even industry that has a number of wonderful scientists that would be very interested about local impacts. So I think that's a good suggestion. I think one thing that the Academy can do is perhaps show the value of this sort of engagement so that other groups feel that it is definitely cost effective to put the time and effort into it. So I've got another question now. And this one is maybe for John, but maybe Karen would want to comment on it too. And that is this concept of just-in-time inventory. Obviously, everyone bought into it because we're involved in this global competition, and just-in-time inventory was the way to be competitive. So if just-in-time inventory clearly got us into trouble with not having stockpiled what we needed for an emergency, how do we get around that, given that we're still embedded in this global competition and any one nation could decide not to do it and undercut American commerce?
So I'm looking at The Economist, but I will toss in my hat into this discussion. You know, from my years in government, part of that is the role of government to look at that. And, you know, under, you know, Janet's leadership and others, we created a national stockpile that was available for response to a bioterrorism event, for response to any major health event. I think we need to reassess that, because in a sense, if we want our businesses to be able to be competitive on the national scale, yet at the same time, there's a national interest in us being able to respond, then there's a logical role for government in making sure that we have that surge capacity to be able to do that kind of response.
So now let's turn to The Economist to see what she has to say.
Certainly right. Supply chain issues have been just, you know, huge during the pandemic, and huge in a way that not only affected kind of just, you know, what we as consumers can consume, as John would say. I mean, they've been huge in our ability to fight the virus. I agree with John that there's a role for government here. I am hoping that it will be taken as a lesson learned. I suspect we will see companies diversify their supply chain coming out of this. I guess part of what makes me hopeful is we did see, you know, we had this terrible financial crisis a dozen years ago, and we did see both companies, but also importantly, government adapt such that we actually didn't see a financial crisis with the pandemic coming upon us. We actually almost, if you were a student of financial markets, you know, we almost did, but the government responded much more swiftly, taking measures they never would have been able to take if we hadn't had the global financial crisis. So I hope it is something I think the government needs to be doing. And I hope it is a lesson taken from this crisis.
Great.
Marcia, let me, might I add a comment to that?
Sure.
First, on just-in-time inventories, one of the downstream impacts is on shipping. And we now have the development of these mega container ships, like the one that plugged up the Suez Canal for a week. We've had to create additions not only to the Suez Canal, but the Panama Canal. The major ports along the eastern seaboard of the United States have had to be dredged and made deeper for the deeper holes. Then you have to have the development of these huge cranes that can unload these containers, et cetera. So I think we're going to have to ask a question in addition to the necessity of having a stockpile, how the just-in-time inventories connect with shipping, and when are ships just too big? to be managed around the globe. And with respect to the stockpile, I would also add that it was a constant struggle to get Congress to appropriate enough funds for a sufficient stockpile. There were some decisions made about what would be in the stockpile. What would be the key crisis that it would need to be prepared for. And so during the years of the Trump administration, they focused on a stockpile in case there was an anthrax attack. Anthrax attack, very different from COVID. And so all things that need to be taken into account in terms of our future resilience.
Yeah.
Marcia?
Yeah. Yes,
Marcia?
Gary. Yeah.
What would happen if critical stockpiles were treated as an accepted and integral part of the nation's infrastructure?
Yeah. Yeah. So
not a side gig, not a thing at the very end that you realize when it's an emergency, but we build a bridge, we build a railroad, we build a stockpile. That stockpile is actually infrastructure.
Yeah. So I think, Gary, the point you're bringing up would be well worth a really in-depth academy study. Because, as Janet just said, you have to know exactly what to stockpile. So figuring out ahead of time what kinds of issues are going to bring you to your knees would be the first step. And black swan things like that ship getting stuck in the Suez. I mean, what kind of person was at the wheel that day? You know, these are huge ships. And he said, oh, my bow got blown into the bank. Okay. All right. So anyway, I think the difficult thing would be figuring out what is needed to be stockpiled. And then also, how do we constantly renew that stockpile such that when we're delivering things, we're taking the oldest out first, sort of like the salad in your refrigerator. You know, you eat the oldest one first. How are we constantly taking the things that are about to spoil or about to go bad or about to be supplanted by new technology and get rid of those so it doesn't become something static that we turn around and say, oh, we've suddenly got this event. Let's go to the stockpile. And you look in the stockpile and there's nothing worth using. So I think
as the national conversation and the bills working through Congress on infrastructure happen right now is the time for stockpiles to be considered in all those conversations.
Yeah. Right.
Gary's making an excellent point that this needs to be viewed as an investment, just like infrastructure is an investment. And with government borrowing costs at kind of rock bottom levels, it just, you know, and now that we understand what's at stake, it does seem like a no-brainer.
So let me turn to another question for you all, and that is the following. Karen, you're involved right now in the rental eviction. Gary, you're involved in the ecosystem services. But what do each of you think? I mean, this is one of those situations where the art in all of this is what question you address. So what do each of you think right now is the next most important scenario that we should be considering in order to be prepared for what are potential cascading consequences of this pandemic?
Karen.
So do you so do you mean within the rental evictions area or just
no, just if you were to pick another topic, not eviction, not ecosystem services. You know, we can't address a problem that we haven't thought about. Yeah. So what do you think is the next one?
I think I'm going to speak as an economist. I think there's been a lot of talk, and I actually agree with this, that our economy in this country is heading, for a recovery this year, a very strong recovery as people are vaccinated. And, now, speaking of stockpiles, people have all these kind of financial resources. They're going to be out there kind of buying, but I don't think we should take it for granted that's going to map into an economic recovery for the entire population. And I think people are just assuming, oh, now we can go back and we can consume services again. All those people, many of whom are kind of low wage workers who have been displaced in the services industry, they're just, they're going to be hired and their jobs are going to come back. And I don't think we can take that for granted. We don't know. We think firms automated during the pandemic, firms whose workers were laid off. So we don't know that these jobs are going to be recreated. We had many small businesses fail. So there's work there to replace them with new small businesses. So I think the thing we should be focused on is kind of what if the recovery isn't shared broadly? I mean, what if many of us experience recovery, but what happens to people and families that were left behind, and what do we do for them?
Okay, great. Thanks, Karen. So we've got a minute each for the rest of you to give me your 1 minute synopsis of what you think is the next problem we need to be addressing. So Gary, are you ready to go?
Yes. I believe that COVID-19 has been an extraordinary, massive, natural experiment in how we do education in the United States, K through 12 as well as higher education. And we need to figure out what are the permanent effects, what are the transitions, and what are the temporary effects of this massive change and disruption on our education system. And then we've got to deal with the outcomes that emerge from that. So I would think the impact of COVID-19 on education and separating out what is transitional from structural and what the outcomes are.
Perfect. All right. Janet?
I think we should ask ourselves, what if there were some way to effectively deal with misinformation, disinformation on social media? What is that, a government issue? Is it a private sector issue? Is there a way to better curate information that is commonly available, and that is causing, I think, and leading to so many people resisting vaccination even now? So dealing with the whole social media Gordian knot, I think, is a worthy endeavor.
I love that one. A topic very near and dear to my heart. Okay, John, we're going to give you the last word here.
Great. And I'm going to delve in. I think that one of the biggest lessons is the impact of decades and centuries of racial inequity in the society has resulted in a disequitable toll upon people of color. And that that we need to look at what are the ways that we can create more resilient communities by having a more equitable society.
That's perfect. So I see Martine Sepulveda, who is the leader of this wonderful scenario planning group. Martine, do you have any final words before I close this out?
Let me just share with everyone who is participating that Marsha's introduction of this particular tool to extend thinking beyond normal processes and methodologies was incredibly well received. The people who you've heard from in this panel and those whom you haven't have taken insights and experiences and started to share those, within their networks. And we would encourage all of you to do the same. Thank you for joining this marvelous panel.
Well, thank you, Martine, for your leadership. And I want to thank all of the panelists for this panel, and actually all of the discussion leaders and panelists that we've had all day. This has truly been a pleasure. I have learned so much. And now it's time to close out the symposium for the NAS annual meeting. Thank you for everyone who tuned in. Okay, and we'll see you tomorrow.